Okay, as we've been discussing, welcome back to the session on nephrology where we'll be discussing on the acid base. So as I told you before, the first thing in nephrology is to understand the physiology. In that matters, we'll start with the acid base analysis. So as we have been discussing on the normal values, the pH normal value is somewhere around 7.36 to 7.44. And the PaCO2, the arterial carbon dioxide normal value is somewhere around 36 to 44 millimeters of mercury. And we have the normal partial pressure of oxygen, where in the arteries, it is somewhere around 75 to 100 millimeters of mercury. But I've told you many times in the past that whenever the values are less than 60, this is what we refer to as something called a critically decreased PaO2. And this is also the definition of respiratory failure. And at the same time, we have normal bicarbonate, which is approximately around 22 to 28 millimeters of mercury. 20 to 28 milliequivalents per liter. If you want to take average, I can take it as 24 as an average. That's what many books will tell. And some people are telling it's trucking. Is it getting stuck in between? Is it clear or is it getting stuck? No. Okay, then Dr. D has to adjust his resolution and internet connection, I guess. Okay, so 24. And as far as the PaCO2 is concerned, it is going to be 40. That's going to be the normal value, 40 millimeters of mercury. Then we can take the sodium value. Sodium value is normal is 136 to 144 milliequivalents per liter. And potassium value is normal somewhere around 3.5 to 5 milliequivalents per liter. And we have chloride values, which is somewhere around 95 to 105 milliequivalents per liter. And we have something called an anion gap, right? Anion gap, which is somewhere around 8 to 12 milliequivalents per liter. But nevertheless, for understanding, you should tell less than 12 milliequivalents per liter is considered to be normal. If you use my formula, and the my formula in the sense the standard formula. Standard formula says that anion gap is equal to sodium minus chloride plus bicarbonate. So this is the standard formula for anion gap. If you ask me why you should not use potassium, if you use potassium, the range will be different. For example, it becomes 16, but that is not validated properly. So that's why for all practical purposes, if you use this formula, then the anion gap becomes less than 12. That is the standard. Then we have another entity called as A gradient. That's again important for your ABG analysis. Everyone knows what you mean by AO2 gradient. So I can write as AO2 gradient also. This should be less than 30 millimeters of mercury. There's a normal A gradient. So if somebody wants to know how will you calculate the A gradient based on ABG, it's quite simple. So it is PaO2 minus PaO2. So we already know the PaO2 because this PaO2 value will be got from the acid-base gas analysis. So the ABG machine is going to give the P small AO2, but everyone will be having a doubt how to find out the P capital AO2. For that, you have the alveolar gas equation. So through which we can find out the partial pressure of alveolar oxygen. So it states that 713 multiplied by FaO2 minus PaCO2 divided by 0.8. So this is standard formula. This will be applicable if you are at sea level. If you are at high altitude, then this will not be applicable. But nevertheless, majority of us are located at sea level. So this formula is what we consider as standard. 713 multiplied by FaO2 minus PaCO2 by 0.8. So this is the alveolar gas equation. So this value minus, if you get the PaO2, you will be getting the AO2 gradient. So then we need to know the definition of type 1 respiratory failure. As you know, type 1 respiratory failure is reduced PaO2 with reduced PaCO2 and probably increased A gradient. So this is the definition of a type 1 respiratory failure. As far as the type 2 respiratory failure is concerned, there will be reduced PaO2 with an increased PaCO2. So this is going to be the definition of a type 2 respiratory failure. Nevertheless, so this is going to be the most important, and all the normal values, whatever it is there, I told you. So the two most important values are the carbon dioxide and the bicarbonate. The average values of 40 and 24, again, it is important. And the third important value is the PaO2 of less than 60, which is called as respiratory failure or critical value of PaO2. And next important value is the anion gap, where I already marked if it is less than 12. So that's considered to be normal. And next important value is the A gradient value. If it's less than 30, we can consider it to be normal. Or some books will tell you less than 12 also, but you can take anything. Less than 30 or less than 12, it's your wish, not a problem at all. And the definitions of respiratory failure should be absolutely proper. And the average pH is going to be 7.4. So how they're getting the 7.4 as normal. So suppose if you plot a normal curve, so the mean value is 7.4. If you take 3 standard deviations away, it will be 7.36. If you take the 3 standard deviations on the other side, it will be 7.44. So that is how they keep this as a normal range. So almost 95% of the people will be having this pH between 7.36 to 7.44, which we consider it to be normal in our setting. So that's why the mean is 7.4. That's why majority of the people will have a pH of 7.4 only. That's why we are taking it as an average in that setting. So nevertheless, so this statistical data, let us move on to the additional analysis. So once you know the statistics properly, now we know about the kidneys maintenance of acid-base balance. Before that, we need to know the concept of something called a buffer. So what do you mean by a buffer? Buffer, I will imagine it to that of a sponge. So for example, if you have a tap that is leaking, and let us assume it is leaking at a very slow rate. So 1 drop by 1 drop, it is getting leaked. And you are keeping a sponge. Maybe you can remember like a duster kind of a sponge it's used in the school. So that's a characteristic sponge we talk about. And because the drop is coming once in 5 minutes, 1 drop is coming. And you're actually, to prevent that 1 drop from leaking into the floor, you're keeping a sponge underneath the tap, which is leaking very slowly. Now you'll be noticing one thing, that after some time, maybe after 1 hour, if you see, there'll be no change at all, which means whatever water drop that is coming from the tap is going to get absorbed by the sponge, and over time it will get evaporated also, because the surface area of the sponge is huge. Once the water is distributed, it will easily get evaporated. So what change you're noticing? I don't think you will notice any change. The duster will be just fine and the floor will be just fine. Still the water will be dribbling from the trap. So this sponge is something which can be equal to that of a buffer. So buffer is something that takes up the changes in the acid-base balance in the body. I mean, any problem in the acid base, for example, if there is excess acid or there is excess base, this buffer is the one that absorbs these changes and then prevents any change in the pH, which means the floor getting wet, I can take it as a pH change. So that will not happen if there is a good sponge that is absorbing all these acid-base changes in day-to-day life. So that's like a buffer. So we have many types of buffers, but of course, if they ask you the most effective buffer right now in the body is going to be our bicarbonate buffer. That's going to be the most important. As far as bicarbonate buffer is concerned, everything is going to work by the Henderson-Hasselbalch equation. We know that. So this Henderson-Hasselbalch equation. So, but more than knowing the Henderson-Hasselbalch equation, it's important because we don't have much time right now. So it's better to know about the Henderson-Hasselbalch rules rather than the Henderson-Hasselbalch equation. It's better to know about the Henderson-Hasselbalch rules. So what does the Henderson-Hasselbalch rules say really? Henderson-Hasselbalch rule says that suppose if there is, there are two things. There will be a pK of the solution. The intrinsic compounds pK will be there, and there is something called a pH. So when you compare these two things, you can derive certain entities. For example, if you take a bicarbonate buffer, so you will write the equation like this. You'll have H2O and CO2, which will unite to form H2CO3, and this H2CO3 will not be stable at all. Immediately it will dissociate into H plus and HCO3 minus. And everyone knows that this reaction is reversible and this reaction is also reversible. But this reaction is reversible only because of one reason, that is because of the presence of carbonic anhydrase. There are different isoforms of carbonic anhydrase that can convert H2O to H2CO3, and which can also convert H2CO3 to H2O, which means we have multiple different isoforms like 1,4, which can do different directions of reactions. So that's what carbonic anhydrase enzyme is all about. But remember, this reaction cannot happen spontaneously. So it needs the help of an enzyme, which means that is not a stable compound. Like in this, they are ultra stable compounds, which means when H2 and CO2 are there, they don't really naturally combine with each other to produce carbonic acid. Similarly, when carbonic acid is formed, they don't really get driven towards the formation of H2 and CO2. So which means these are not naturally mediated reactions, which means that should be done by an enzyme. That's what carbonic anhydrase is going to do. So naturally, they don't combine with each other, or naturally H2CO3 does not dissociate into H2 and CO2. Instead, This reaction is where you need to know about the Henderson-Hasselbalch equation, because this reaction is basically based on the pH and the pK of the solution, which means this is an equilibrium reaction. There's a difference between equilibrium and enzyme-mediated reaction. This is spontaneous equilibrium-mediated reaction, which means once H2CO3 is formed, depending on the pH and the pK of the solution, H2CO3 can remain as H2CO3, or it can dissociate into H plus and HCO3 minus. So this is something that is driven by the Henderson-Hasselbalch equation, because that's a spontaneous equilibrium-related reaction. But the other side, which I've told you, is not possible to happen. That is an enzyme-mediated reaction. Both are completely different. So which means this particular side can be equated towards our Henderson-Hasselbalch equation. For example, based on the pK and the pH of the solution. What Henderson-Hasselbalch rules says is, if the pK is less than that of the pH, what will be the amount of dissociated and undissociated forms? If the pK is equal to pH, what will be the amount of dissociated and undissociated forms? Or pK is more than pH, then what will be the amount of dissociated and undissociated forms? For example, I can say that this H2CO3 is considered as an undissociated form, and this area, this H plus and HCO3, can be considered as dissociated forms of H2CO3. The rules are clear that if pK is equal to pH, Henderson-Hasselbalch rules state that the dissociated forms will be equal to the undissociated forms, which means amount of H2CO3 molecules will be equal to that of the molar concentration of H plus and HCO3 minus. Suppose if the pK is less than the pH, then the amount of dissociated forms will be more than the amount of undissociated molecules, which means amount of bicarbonate will be more than the amount of carbonic acid. That's what this pK less than pH relationship is trying to tell. If pK is more than pH, then in this scenario, the amount of undissociated forms will be more than amount of dissociated forms. That's what the equation is telling. So let us assume that the pK of carbonic acid under our routine conditions is 6.1. pK is 6.1. So let us assume, even though at 25 degrees it is 6.4, at 37 degrees in body temperature, it's somewhere around 6.1. So let us assume this is the pK of carbonic acid, that is H2CO3. So now, according to the Henderson-Hasselbalch equation, so what do you think it should happen? So here the pK is less than pH, or pK is more than pH, or I wanted to get an answer that what will be the correct among the three. The dissociated forms will be more than undissociated forms, or undissociated forms will be more than dissociated forms, or dissociated forms is equal to undissociated forms. So what will be the equation balance, and what will be the number of dissociated forms according to the Henderson-Hasselbalch equation? You're going to answer. So I think there are a sizable number of people are there. Yeah, according to the Henderson-Hasselbalch rules, of course, the dissociated forms should be more than the undissociated forms, because here the pK is less than that of the pH. Clear? So that is the reason why, because the normal pH, for example, we know the normal pH is 7.4. So we know that. So the pK is lesser than that of the normal pH. So according to the natural Henderson-Hasselbalch equation and the rules, the bicarbonate should be more than the carbonic acid forms. So that's very true, because in our body, normally the amount of bicarbonate that we see is 20 times more compared to that of the, I can write CW compared to that of the carbonic acid. So hence proved, because the bicarbonate, why it is higher, because according to the Henderson-Hasselbalch rules, the pK is less than that of the pH, then the dissociated form should be more than undissociated forms. That is why naturally bicarbonate is more, at most 20 times more than that of the carbonic acid. You might ask why bicarbonate is in higher amount in the body compared to that of the carbonic acid. Because our body always has a tendency to produce more acids, in the sense like to metabolism, we are going to generate some form of acids called as fixed acids, or new acids can be added at any point of time. So to neutralize this acid, definitely we need more bicarbonate. So that is the reason why, one of the reasons why body tends to really use this bicarbonate buffer effectively and tends to have more bicarbonate than the carbonic acid. Point number one. So that is the reason. Apart from that, this excess bicarbonate, 20 times more bicarbonate is there compared to carbonic acid. This excess bicarbonate that is naturally present in the body is what we refer to as something called as a bicarbonate reserve. So why we need that reserve? Whenever in times of acidosis, which the body is generally prone to develop. So usually your bicarbonate will rush into the rescue because that reserve bicarbonate will come and they will neutralize the acids and they will, neutralize any change in the pH of the solution that is happening. In this case, it's the blood. You can have a second example in this scenario. The second example is ammonium. So there is one compound called ammonium. Ammonium dissociates into ammonia and H+. We know that. So if you ask the pK of the ammonium, if you take the pK of the ammonium is 9.4. So which means it is definitely less than that of the pH. Sorry, it is more than that of the pH, which means the body's pH is 7.4. It is definitely more than that of the pH. So as far as the ammonium is concerned, it will be in the dissociated form or undissociated form. According to our Henderson-Hasselbalch rules, it must be in dissociated form or it must be in the undissociated form. Definitely, according to the rules we follow, it must be basically in the undissociated form. So that's what happens in the body as well. At a body's pH, ammonium tends to exist in ammonium form only. It does not dissociate into ammonia and H+. So ammonium is something that tends to exist and that they want to exist in the ammonium form only. And this becomes really important when we want to understand the renal buffers in the form of ammonium buffer. That time it will be very helpful. Okay. Now, having understood about the concept of this Henderson-Hasselbalch equation and the buffer system, why it is actually existing, this is to neutralize any change in the acid-base balance that is happening in the body and to maintain the pH of the blood or pH of the solution in the normal range. That's why we have these buffer systems in place in the first place. So after this, we need to know the kidney's role in understanding the acid-base regulation. But please understand, there are two organs which are quintessentially important in maintaining acid-base balance. One is the lung and second is the kidney. So this is the only buffer. For example, your bicarbonate buffer is the only buffer that has connections with both. For example, through carbon dioxide, they have connections with lung also. And through bicarbonate and H+, they have connections with the kidney also. Which means this is the only buffer that has connections with two different organ systems. Based on they can regulate the acid-base changes in the body. So you might be asking, so for example, there are something called volatile acid in the body. So if you ask, what do you mean by volatile acid? I always tell this CO2 as a volatile acid. And if you ask, what do you mean by a fixed acid, whenever there is an increased load of H+ in the body, we call them as fixed acids. But nevertheless, you can ask how CO2 is considered as a volatile acid in the first place. You can understand there is something called carbonic acid. With the help of course, carbonic anhydrous, they can form H2O and CO2. And H2O contains H+ and HCO3-, that is bicarbonate. Many people do argue telling that, sir, chemically equation is balanced, but how can you say that CO2 is considered to be an acid in the first place? Because water, we know it's supposed to be a neutral compound. But how come this CO2 can be an acid in the first place? People do get, because this H+, how when it becomes CO2, it becomes an acid. So for that, you need to understand the concept of protons. So what do you mean by acid? Acid is nothing but a proton. That is what we refer to as an acid. That's why whenever you write a pH, always they tell it is inversely proportional to the H+ concentration, which means whenever H+ is increasing, pH is decreasing. Whenever H+ is decreasing, pH is increasing, which means H+ is the one that is considered as an acid that is going to reduce the pH of the solution. This is a very simple concept. But for people who don't know that what we are talking is about the proton. For example, if you take a hydrogen atom, it's the simplest atom in the universe and the first atom to form in the universe. There is one proton and one electron. That's all. This is hydrogen atom. Suppose if you knock off this one electron from the hydrogen atom, there will be only protons that is left. So this is what we refer to as H+, which means this is what we refer to as something called as an acid, which means acid is nothing but a proton. That's all. A proton, if it's in excess, that's what we refer to as an acid. To be honest, this CO2, even though it is hidden, it carries protons with it. CO2 is the one that carries that protons because H2O is neutral. This proton that is present here in this carbonic acid will be actually carried by the CO2 only, because CO2 is a protonated form of the molar compound. So that is the reason why CO2 is considered as a volatile acid in the first place, which means elimination of CO2 through the lungs is equal to elimination of protons through the lungs. Elimination of protons by any form outside the body is considered as elimination of acid outside the body. That's why CO2 elimination is basically equal to that of the acid elimination. So lungs are something that are very, very important in eliminating the acids. For example, more than 13,000 millimoles of carbon dioxide is getting eliminated per day, which means lungs play a huge role in maintaining acid-base balance by eliminating a lot of tremendous amount of acids in the form of carbon dioxide. That's why carbon dioxide is supposed to be called as something called a volatile acid. Similarly, bicarbonate and H+, the fixed acids are basically regulated by the kidney. So volatile acid is regulated by the lung. The fixed acids are basically regulated by the kidney. So now, having said about the lungs' role in regulating this carbon dioxide and volatile acids, now it's important to know about the kidneys' role in regulating these fixed acids, how the kidneys regulate the acid-base balance. There are two areas where they regulate acid-base balance. One is going to be the proximal convoluted tubule, another is the distal nephron. So two areas I can draw side by side. One is the PCT, which regulates the acid-base balance, and second is the distal nephron that still regulates the acid-base balance. So how will you really appreciate this PCT's role and distal nephron's role? Because both are going to be very important in day-to-day physiological settings, but which is more important in the setting of, acid-based disorders, your PCT or your distal nephron, which is very important in the setting of acid-based dysregulation or in the presence of acidosis or alkalosis? Which area is very important in regulating the acid-base balance, proximal convoluted tubule or the distal nephron? Which is more important? Many students are actually answering about the proximal convoluted tubule, but I do believe that it's the distal nephron that's the most important. Distal nephron means it includes the collecting duct. That's the most important, not the DCT, and the most distal part of the DCT. The most distal part of DCT is also important to some extent. So apart from that, the collecting duct is where my main role of acid-base regulation is left. So distal nephron basically is important for three reasons. One is your acid-base regulation. Second is potassium homeostasis, and third is urinary concentration. These three functions, even though many other areas are also doing these functions, I'm not telling no. But when it comes to problems in the body, the distal nephron is the one that takes the primary place in controlling and changing these things in the body, like acid-base balance, potassium homeostasis, and your urinary concentration. These are three things that where distal nephron is extremely important, because distal nephron is the site of ADH action also. That's why urinary concentration and the subsequent urine osmolality and plasma osmolality will be depending on. And at the same time, distal nephron is also the main entity for the acid-base regulation in the times of acidosis and alkalosis, especially. And at the same time, distal nephron is important for potassium homeostasis as well as the setting of hyperkalemia and hypokalemia. So let us start with the PCT. Why PCT is not that important, but in the sense like PCT is also important. I'm not telling relatively why it is not that important. But let us see what are the function of any tubule. So there are, kidneys are divided into glomerulus and tubular segment. We know that. Kidneys are basically the ultrastructure is divided into glomerulus. The nephron is divided into glomerulus, and we have the tubules. So as far as the glomerulus is concerned, we know the most important function is the filtration function. So which means they are going to produce the glomerular filtrate, GF. That's the glomerular filtrate. And as far as the tubules are concerned, the tubules have many different functions. The most important function is something called a solute reabsorption, which I'll be talking about that in AK as well. And second, they have something called acid-base regulation. And the third function is urinary concentration. Apart from that, they have multiple other functions. For example, for vitamin D synthesis, active vitamin D synthesis, then many other functions, ammonia genesis, many other functions they do. But these three are the primary functions of the tubule: solute reabsorption, acid-base regulation, and urinary concentration. So these three are the most important as any nephron. So these are the things they are going to do. So if they ask you about the solute reabsorption, so let me tell you the basics here itself. Whatever may be the fact, the maximum solute reabsorption for almost all the solutes is actually the PCT only. PCT is the one that absorbs more than 60 to 90% of all the solutes whatever may be the fact, but exception of two solutes, that is calcium and magnesium. Except calcium and magnesium, all the solutes are maximally absorbed in the PCT, including water. Maximally absorbed is PCT only, except calcium and magnesium. Calcium and magnesium are predominantly absorbed in the thick ascending limb of the loop of Henle and probably in the distal convoluted tubule. So to an extent. So they are primarily absorbed in the thick ascending limb of the loop of Henle and the distal convoluted tubule, DCT. Apart from that, all other solutes are predominantly and maximally absorbed in PCT only. In fact, almost 100% of the glucose is absorbed in the PCT only. Almost so much of 65% plus water is absorbed in the sodium and water is absorbed in the PCT only. So solute reabsorption, PCT plays a very, very important role. For acid-base regulation, you need PCT as well as DCT. Sorry, PCT as well as the distal part of the nephron. You should not write DCT. So the distal nephron, especially the collecting duct portion, is very important. Even though in times of need, the distal nephron is the one that comes into our rescue. As far as urinary concentration is concerned, all the areas are important, but I will feel that the thick ascending limb of the loop of Henle and at the same time your distal part of the nephron, that is the collecting duct especially, again. So these two are the most important. These two are equally important. I mean, in a nutshell, this is what kidneys do. So when you take about the PCT, so PCT does a variety of functions. So let us see the first function, that is solute reabsorption. So as I told you before, second one, they does something called that acid-base balance also. In the setting, they do something called the bicarbonate reclamation. I'll talk about that in some time. What do you mean by bicarbonate reclamation? And third thing that PCT cells do is they are going to produce active vitamin D, which means they have an enzyme called 1-alpha hydroxylase enzyme. This 1-alpha hydroxylase is the one that converts this calcidiol, that is 25-hydroxycholecalciferol, to 1,25-dihydroxycholecalciferol. So that is the active form of vitamin D, that is calcitriol. Clear? So second function. A third function, that is 1-alpha hydroxylase activity. And fourth function is they do something called ammoniogenesis, which we'll be talking about in detail. Ammoniogenesis is a very important function that is done by the proximal convoluted tubule. All right. So these are the four primary functions that we don't want solute reabsorption right now. We don't want this function right now because we are not going to discuss in detail about that because we're talking about an acid-base balance. So the bicarbonate reclamation and the ammoniogenesis is going to be more important in this particular example. So let us see how they do that. So and why we call it as a bicarbonate reclamation in the first place. For that, we need to understand what's really happening in the tubule. So first, let us see the proximal tubule here. And we know that this is the afferent arteriole and this is going to be the efferent arteriole. And I'm drawing the PCT cell here. PCT cell here. So once the PCT cell, let us assume there is some amount of filtered bicarbonate. So how much will be the filtered bicarbonate will be based on the serum bicarbonate. For example, if you want to know the filtered fraction of any compound, that is the filtered bicarbonate, that will be equal to the glomerular filtration rate into the serum concentration. In this case, it's the serum bicarbonate. So we know what is the glomerular filtrate per day. Per day, the glomerular filtrate is 180 liters approximately. If you calculate 125 ml per minute, then for 24 hours, it will be 180 liters multiplied by the bicarbonate, that is 24, which is average. We saw already serum bicarbonate 24, you will end up around 4,320 milliequivalents of bicarbonate. So I can tell average around 4,000 to 5,000 milliequivalents of bicarbonate that is getting filtered. So this is the filtered bicarbonate that we are talking about. Around 4,000 to 5,000 milliequivalents of bicarbonate is getting filtered every day. And who's going to absorb all this bicarbonate? Around 80 to 85% is absorbed by the PCT, and approximately 10 to 15% is absorbed by the distal nephron. That is especially the collecting ducts we are talking about. So which means for reabsorbing this filtered bicarbonate, your PCT is extremely important. Extremely, extremely important. How they reabsorb? Sorry for that, reclaim this bicarbonate. They don't reabsorb this bicarbonate directly. Instead, there is a series of processes that will happen. Initially, this bicarbonate will combine with something called H+. Let me zoom a little bit and write. So there is something called H+ that will be coming out here through a transporter called as sodium H+ transporter. This is what we refer to as something called a NHE3, sodium hydrogen exchanger we are talking about. So H+ will come out and this bicarbonate is going to combine with this H+. And of course, they're going to form H2CO3. But remember, since the pH of the solution is at this point is actually 7.4. So if the pH is 7.4, then according to the Henderson-Hasselbalch equation, you know what is going to happen according to Henderson-Hasselbalch equation. We saw already that by the moment the carbonic acid is formed, immediately it will result in dissociation into H plus and HCO3 minus. But if that happens, then there is no point in having this whole process in the first place, and why they want to excrete this H plus outside. So this will not happen because in the brush border, there is a very important enzyme called carbonic anhydrase. This is called brush border carbonic anhydrase. That is in the urinary surface, outside in the lumen of the tubules. So this carbonic anhydrase will trap this carbonic acid that is formed, and they will convert to H2O and CO2. So it is assumed that this CO2 and probably H2O is taken back into the cell, and inside the cell you will have, of course, H2O, or you can take the H2O and they will combine together. And once again, this combination is not possible unless and until you have a carbonic anhydrase. So inside the cell also you will have carbonic anhydrase, and this is going to convert this H2O and CO2 into H2CO3. And of course, once this forms, because this isoform is a different isoform, they can do only production of H2CO3, and they cannot dissociate H2CO3 into H2 and CO2. So automatically, according to the pH, this H2CO3 must dissociate into H plus and HCO3 minus. And this HCO3 minus will be taken by separate chloride bicarbonate antipode, where this chloride will be moving away through separate chloride channels. And this is how bicarbonate is reclaimed, basically. So this is a process that you should not call it as a reabsorption, because there is no direct reabsorption that is happening. So if the bicarbonate is directly reabsorbed just like that, then it will be called as a reabsorption. But this is not a direct reabsorption. It is an indirect way of absorbing bicarbonate back into the body. And this is what we refer to as something called a bicarbonate reclamation. That's why textbooks will give you reclamation. It's not a reabsorption. It's like a magic, isn't it? Bicarbonate is coming down, but after a series of steps, bicarbonate is reclaimed back. The same bicarbonate that is here has come here now. So it's happening like sort of a magic, but still, it's happening. So this is what we refer to as bicarbonate reclamation. And this is a very important process. Almost 80 to 85% of the bicarbonate is basically absorbed in the proximal convoluted tubule through this particular reaction only, which is very, very important in our understanding. So for effective reabsorption of bicarbonate, you need to actually pump out this H plus. Pumping out this H plus is also important, because even though you can argue that this H plus is the same thing that is coming out again and again, which means this H plus comes here, somehow through magic, through carbon dioxide, through CO2, this H plus somehow goes inside, because I told you CO2 is the one that captures this proton. So that's a protonated form. So CO2 goes inside, and as a magic, again, this H plus is regenerated, and again, this H plus is coming out through CO2. Once again, it goes inside, it comes out. So which means it's sort of a getting recycled over at this point. So if H plus is getting recycled, you can argue that the amount of H plus that is actually secreted by the PCT is quite less, which means the same H plus is getting recycled again and again and again. So which means what you can tell is the amount of H plus that is actually getting excreted through the urine by the proximal convoluted tubule is actually not that great. That is why this NHE3 transporter, this is sodium hydrogen exchanger, is referred to as something called a low gradient pump. So why it's called a low gradient pump? Because the pH change will not be that great in the setting of a NHE3 pump that is present in the PCT. And that's a very important point, because they cannot change the pH of the urine dramatically. If the pH of the urine is 7.4 at the time of the entering into the PCT from the glomerular filtrate, at the time it goes down the PCT and crosses the PCT, it will be maybe around 6.9 or 7. So there will be not much change in the urinary pH. That's what I'm trying to tell. So that's why it's a low gradient pump, because the pH H plus is not effectively secreted here. H plus is just recycled again and again, more than getting secreted permanently into the urine. So that's one thing. But this pump is often referred to as high capacity pump. Remember, for every 1 molecule of bicarbonate to get reabsorbed, you need to secrete 1 molecule of H plus in this particular area. So if you want to secrete 1 molecule of H plus in this particular zone, you can argue that for reabsorbing this 4,000 to 5,000 milliequivalents of bicarbonate, you need to actually secrete 4,000 to 5,000 milliequivalents of H plus as well. So unless and until you secrete the same amount of H plus, the bicarbonate will not be reclaimed or reabsorbed, and they will be excreted in the urine. So to have an effective bicarbonate reclamation, your H plus secretion should be happening in 1:1 ratio. Since PCT is the one that absorbs 80 to 85% or reclaims 80 to 85% of the filtered bicarbonate. For example, if PCT is the one that absorbs, example I'm telling, PCT reclaims 4,000 milliequivalents of bicarbonate. Let us assume 85% of 5,000. We can assume approximately 4,000 milliequivalents of bicarbonate. To do that, they have to secrete 4,000 milliequivalents of H plus as well, which means the secretory capacity is extremely high. So that is why this is also referred to as a high capacity pump. So this PCT related NH3 transporter is also referred to as a high capacity but a low gradient pump, because the effective change in the pH will be negligible, but at the same time they can secrete high amounts of H plus throughout the day, 24/7. It's a workaholic, and they can effectively reabsorb bicarbonate 24/7. So much amount of bicarbonate is reabsorbed by the PCT, proximal convoluted tubule. Okay. So now you would have understood how this reclamation mechanisms happen. But now let us move on to the distal convoluted tubule or distal nephron. So as I told you, the distal nephron's most important function is to maintain acid-base balance. And the second most important function is potassium homeostasis. And the third most important function is your urinary concentration. So these are the most important functions of the distal part of the nephron. That acid-base function plays a very, very vital role and important role. Let us see what distal nephron can do and what are the cells in the distal nephron can do. So if we take a distal nephron, let me draw a little bigger. So this is a tubular lumen, and everyone will be knowing what you mean by apical segment, what you mean basolateral segment. So this particular area where the urine is touching the cell, this is what we refer to as a luminal side or a apical segment. And this area where it is touching the capillaries, is what we refer to as a basolateral segment. So whatever I've written in red are basolateral zones or basolateral segment. Whatever I put in green here are apical segments, apical segment of the cell. So now you know what you mean by apical or luminal and what you mean by basolateral side. So whenever you study in textbooks, this should be very clear first. What is apical and what is basolateral? Now, as you move towards the distal part of the nephron, there are two types of cells are there in the distal part of the nephron, especially in the collecting duct. One is going to be the alpha intercalated cell. This is the first one that is important. And second one is the beta intercalated cell. The second cell that is important. So this is a beta intercalated cell. And before that, you have something called a principal cell. I think I can redraw this. So this is the principal cell. I can write as a P cell, and this is an alpha intercalated cell, and this is going to be the beta intercalated cell. So for time being, you can understand why it is called a principal cell, because principal cell is the principal site of aldosterone action. Number one, that's that area where aldosterone acts typically. So that's why it's called a principal cell. And then importance of the alpha intercalated cell is the fact that it's important in times of acidosis. During acidosis, this is the cell that becomes extremely important for our body. And the beta intercalated cell, you can think that in the times of alkalosis, this beta intercalated cell is the one that becomes really, really important for our body. So these are the three zones that we can talk about: P cell, alpha intercalated cell, and the beta intercalated cell. P cell is important in terms of aldosterone action, and our alpha intercalated cell is important in terms of in times of acidosis, and beta intercalated cell is important in times of alkalosis. So as far as the P cell is concerned, how the aldosterone really acts in the P cell. So let us take this into perspective and let us see. We know that thing very clearly. So first, aldosterone is a mineralocorticoid hormone which has its own receptors. So let us assume you have an aldosterone receptor here, where aldosterone binds in the aldosterone receptor. Once they bind, they come and attach to the nucleus, in the sense in the certain DNA segments that are responsive to mineralocorticoids, and they're going to increase the transcription of certain proteins. And there are two proteins that will be increased in number and function. One protein is going to be the epithelial sodium channels. We know that these are ENaC channels or epithelial sodium channels, which are going to be increased in number. At the same time, there is another thing that will also be increased because of this aldosterone action, that is sodium potassium ATPase in the basolateral side. So which means aldosterone action increases the number of epithelial sodium channels in the luminal side and the sodium potassium ATPase in the basolateral side. So basically what's going to happen is very simple. You are going to reabsorb a lot of sodium. This sodium will be moving out through the sodium potassium ATPase. That's why they are doing both. And at the same time, what happens is your potassium that comes in, this potassium anyways will accumulate within the cell. But because of the movement of the sodium back into the cell and into the basolateral side, this will create a strong negative potential on the luminal side of the membrane, on the apical side of the membrane. That is because of the movement of sodium into the cells. And this will actually create an electrochemical gradient that will suck the potassium outside through these channels called as ROMK potassium channels. That's called ROMK, renal outer medullary potassium channels. And this is something which is well known under very basic physiology. So which means aldosterone facilitates the movement of sodium in the body, which means they reabsorb sodium. At the same time, they also facilitate the movement of potassium into the lumen, which means potassium will get wasted. So the primary effect of aldosterone is very simple. They are going to increase sodium reabsorption and they're going to increase the potassium secretion. But the primary effect, if you ask, it will be the sodium reabsorption. And because of that only, your potassium reabsorption is happening. Potassium secretion is happening. So this is the primary effect of aldosterone. So that is why whenever you have a hyperaldosteronism, you will result in development of hypertension because of excessive sodium reabsorption and hypokalemia because of excessive potassium secretion. Whenever there is a hypoaldosteronism, you will get hypotension and hyponatremia, and there will be salt wasting because sodium cannot be absorbed properly, and there can be hyperkalemia. Even though not always true, but this is something like a very basic level understanding, aldosterone action. Now, this potassium that moves in has a lot of other effects. But before that, let us see what are the functions of alpha intercalated cells. In the alpha intercalated cells, there is one special function that is called carbonic anhydrase function, which is located de novo in this alpha intercalated cells, which means they can take up this H2and CO2 formed by their own intrinsic metabolism, and they can directly convert them to H2CO3 using carbonic anhydrase. This is a special function of alpha intercalated cell. And once you have formed this H2CO3, they are going to dissociate easily into H plus and HCO3 minus, and they can reabsorb this HCO3 minus through the same chloride bicarbonate antiport. And chloride will be moving back into the basolateral side through separate chloride channels. So we not talk about that. So what's been done here is there is no bicarbonate reclamation, which means the alpha intercalated cell has a special ability, and they can form carbonic acid by themselves using this carbonic anhydrase activity that is present inside the cell only. And they can actually eliminate H plus, and they can take up bicarbonate as well, and they can produce new bicarbonate as well. Remember, this bicarbonate that is produced is a new bicarbonate, and this H plus that is secreted by the alpha intercalated cell is basically something that is excreted into the urine. Basically, that is excreted back into the urine. So that's that's the function of the alpha intercalated cell, which means that is why I told you in times of acidosis, it becomes really, really important. In acidosis, you will have excess acid and low bicarbonate in the body. So in acidosis, you make sure that you produce more bicarbonate and you excrete excess H plus in the urine. So that's why this is very important in times of acidosis. And this excretion of H plus happens through two key molecules. One is the characteristic potassium H plus ATPase, which tends to absorb potassium and release H plus. So this potassium H plus ATPase. And there is a second way of eliminating this H plus. Second way of eliminating this H plus, that is the H plus ATPase. There is two types are there, H plus ATPase and K plus H plus ATPase. Both can eliminate H plus in this setting. But even though H plus ATPase does 85% of the function, K plus H plus ATPase does only 15% of the function. So that's what happens in this scenario. So which means the most important reason for elimination of this H plus is the H plus ATPase that is present in alpha intercalated cell, and to an extent done by the K plus H plus ATPase. Now what really happens is this K plus that is eliminated through aldosterone action in the P cell now will act as a stimulus for this transporter, that is H plus K plus ATPase. So what will happen is there will be more potassium. I mean, even though it's a low capacity pump, which means they can absorb only 15%. So they cannot absorb all the potassium that is coming from the principal cell, but still they can, what they can trigger is they can cause this pump to get activated and they can produce more and more absorption of potassium. But even though they cannot collect all the potassium that is wasted by the P cell, some amount of potassium will be absorbed, but still what they can effectively do is they can eliminate more H plus through this particular area. So that is why aldosterone action is going to increase H plus secretion as well through the intercalated cell. And that is the reason why whenever there's an excessive aldosterone levels, you will get metabolic alkalosis because of increased H plus wasting, and hypoaldosteronic conditions can result in metabolic acidosis. That is the reason. So of course, the primary effect is the sodium reabsorption through ENaC channels, and that's resulting in other secondary effects like increased potassium secretion and increased H plus secretion. Now, this is the link between the aldosterone and the alpha intercalated cell. And this is the link between the P cell and the alpha intercalated cell in the first place. Okay. So now we know what are the things that are happening in this sort of a scenario. And we know the importance of the alpha intercalated cell, which is very important in the setting of acidosis. On the other hand, what is the capacity of this alpha intercalated cell? This alpha intercalated cell is extremely important in eliminating the fixed acids. Eliminating fixed acids. So what do you mean by fixed acids? Fixed acids are something that are produced by day-to-day, day-to-day metabolism. Usually the normal amount of acids that are produced by the body is approximately 1 milliequivalent per kilogram per day. This is a normal amount of H plus generation, which means this is a normal amount of fixed acids that are produced in the body by various metabolic events that are happening in the body. And the average, if you take, it will be approximately 70 milliequivalents per day. In a 70 kilogram man, it will be approximately 70 milliequivalents per day. That is the amount of H plus that is getting generated every day. And to eliminate all these fixed acids, you need this alpha intercalated cell, which means you need the distal nephron. So you can ask me, what about PCT? PCT eliminates, 4,000 milliequivalents of H plus, but still they don't have any effect in changing in the pH, because all these 4,000 milliequivalents of H plus that is secreted into the lumen is used for bicarbonate reclamation. That is, they are used for reclaiming back this bicarbonate. They are not actually eliminated out of the system. Instead, they are recycled, and that is used only for bicarbonate reclamation. But here, what's really happening is they are eliminating H plus into the urine, and whatever extra amount of acids that are produced in the body in day-to-day metabolism is actually eliminated by the distal nephron only. And that is why the distal nephron becomes very important for eliminating the fixed acids that are produced in day-to-day metabolism. If this is not working, and again the acids will start accumulating and you will die over a period of time. So that's where the problem is. Suppose if you ask me in times of acidosis, can they expand or not? Of course they can expand in times of acidosis. So suppose during the times of acidosis, they can expand their capacity by 10 times compared to the physiological baseline, which means their acid excreting ability will become approximately 700 milliequivalents per day. 10 times they can expand their acid excreting capacity. But still, compared to the PCT's ability to excrete H plus, this is much lesser. PCT can excrete 4,000, 5,000 milliequivalents of H plus per day to reclaim that bicarbonate. But, your distal nephron can excrete only 700 milliequivalents per day. But still, this is really, really important, because PCT just does a function of bicarbonate reclamation. But here, the actual amount of acid that is generated in the body is getting eliminated through the distal nephron only. So that's why the distal nephron is more important. Even though it eliminates lesser amount of acid, it's a low capacity pump compared to the PCT. So it's a low capacity transporter here that is located in the PCT. Distal nephron compared to that of the PCT NHE transporter. On the other hand, it will be a high gradient pump. So why it's high gradient? Because it can effectively reduce the pH of the urine, which means, for example, the normal pH of the urine is 4.5 to 6.5. So there will be no much change in the urine because of the, and much change in the pH of the urine because of the PCT's excretion of H plus, but the pH of the urine is going to drop dramatically, and the urine is going to become more acidic as far as the distal nephron is concerned. So which means, one more point I can add is this distal nephron is a very, very important area, not only for acid-base balance in the body, it's also very important for acidification of the urine. So this distal nephron is the one that acidifies the urine, and this distal nephron is the one that eliminates the fixed acids out of the system, which means the day-to-day acid production in the body by metabolism will be eliminated by this distal part of the nephron only. So that's why this becomes really important in this particular setting. Okay, fine. So the minimum urinary pH that you can achieve is 4.5. And usually it will be the normal range is 6.5, but the maximum urinary pH can be 7, 8 also. Under certain settings, if forcibly alkalinize the urine, then it can reach 7, 8 also, doesn't matter. But the minimum urinary pH that you can achieve by your distal nephron is 4.5 only. This is the minimum urinary pH. Urine pH cannot become lesser than this. 4.5 is the least, because that much is only the capacity. I mean, even though it's a high gradient pump, there is a limit for that gradient, how much they can eliminate. So till the urine pH reaches 4.5, they can eliminate. That actually gives rise to another question, another important question, that suppose if your urinary pH has reached a minimum of 4.5, which means there is so much of H plus concentration. Let us assume a separate diagram here that will make you understand one more important entity here. Suppose if you want to eliminate H plus through the distal nephron, we are talking about the distal nephron now. Over time, what will happen is the H plus will start getting accumulated here. H plus will start getting accumulated here over a period of time, which means it's like a bathroom. So, I mean, let us assume, not like a bathroom, let us assume an auditorium. So people are inside the auditorium, and there are 2 doors in the auditorium. So 2 doors are there in the auditorium. Through one door, people are keeping on entering into the auditorium. So they are coming, and they're getting accumulated in the auditorium. So over a point of time, you cannot push any more people, because now that's what we call it as a loss of gradient, isn't it? Once the gradient is high, auditorium is empty, outside a lot of people are there, so naturally they move towards the empty place. So then that's fine. So over a period of time, once the auditorium becomes extremely congested and full, and the movement will reduce, and after a period of time, the movement will completely stop. So to avoid this congestion, so what you have to do? I mean, simple question I'm asking, because I need you to be attentive. That's why I'm asking this question. To avoid this kind of a congestion, what you should do? This sort of a congestion. To continuously engage people back into the auditorium. So what you should do? You can imagine like a marriage hall. Okay, Pranav is telling, close the door. Okay. Okay. Yeah, Pranav is telling close the door, and that's not the right option, because these people have to move, because you're talking about an acid, and that it has to go out. Without moving out, I don't think that's correct. So they have to move out, by the way. Okay, fine. So to do this, you need to create an exit. So which means open this door also. So which means as you keep, moving these people out, these people can keep coming in. So that is the idea. This is a simple option. That's what happens in marriage halls. Marriage hall may be like capacity of only, 500 people or 1,000 people hall. But you invite 2,000, 2,500 people. How you invite that many people? Because people keep coming in, keep going out. So that's like a sort of, like a queue. That's what you need to make. So you need to open this door. So that's the most sensible idea. Open this door and keep moving the people out. So let me tell you that these many people are not able to move out that fast, which means they have to actually get moved very fast, because let us assume there are 1,000 people over here, and the auditorium's capacity is only 50 now. Auditorium's capacity is only 50. At one point of time, there will be 50 people that are coming here inside, and 50 people are staying, and somehow you're trying to move this 50 people out. So which means it's a very slow process. So, you have to only clear off 50 every time. And it takes a long time for this 1,000 people to get eliminated. So let us assume you need 1,000 people to get eliminated, but at a time, auditorium can accommodate only 50 people. So why that 50 people limit is there? That you can imagine like a pH of the urine. So minimum pH that you can achieve is only 4.5, which means beyond that you cannot accommodate people, which means it is like an auditorium that can accommodate only 50 people, and which means it's like reaching a pH of 4.5. We cannot achieve a pH less than that, and you cannot accommodate more and more H plus in that. So somehow you need to eliminate these people faster. Suppose let us assume you want to eliminate by flushing the urine. For example, let us take urine. Urine is a natural substance that flows down into the urinary tract, and it is going to clear off the urine. clear off this H+. This H+ can be cleared off just because urine is flowing. Just because urine is flowing. So it's like a kind of a security that is present in many temples and churches, so where they keep pushing you out, isn't it? So you can imagine like that. So if you want to use the security to do that, suppose, and it's like a urine that is keeping pushing out, then it's not possible again. Why? I'll tell you, because let us take a simple calculation. Suppose if you have 1 liter of urine flow, 1 liter of urine flow will be able to eliminate approximately only 0.03 milliequivalents of H+. And this is a equation. This is a straightforward equation. So under these circumstances, it's been derived that 1 liter of urine flow will be able to eliminate 0.03 milliequivalents of H+, natural urine flow. That's how it's going to happen. Suppose if I wanted to eliminate 70 milliequivalents of H+, if you want to eliminate 70 milliequivalents of H+, how much is the amount of urine that I need? So it's approximately 70 by 0.03. That is the amount of urine that I need to eliminate this much amount of H+. That is normally produced in the body daily. Normally we produce 70, and to eliminate 70, you need 270 liters of urine. 270 liters of urine. If urine flow is the only thing that wanted to eliminate, get rid of all this H+ out, and to eliminate 70 milliequivalents of H+, you need 270 liters of urine to excrete just 1 day's acid production in the body. Are you producing this much amount of urine per day? I don't think this much amount of blood itself is there in the first place. Of course, we don't produce this much amount of urine, 270 liters of urine. We produce only 1 to 1.5 liters of urine, or maybe 2 liters of urine per day. So there should be some other way to eliminate this 70 milliequivalents of H+, and not just by urinary flow, which means urinary flow alone is absolutely going to be extremely ineffective method to eliminate this amount of H+. So, I mean, it's coming as 2,700 liters. That's what he's telling. Okay, maybe, whatever may be the case. So to eliminate this much amount of, that much amount of urine you don't produce really. You produce only 1 to 2 liters of urine. And of course, that's not fair. And you should have some other mechanism that can actually eliminate H+ in a very effective way. So that's why we have this kind of renal buffer. So which means the urinary flow alone cannot maintain this amount of H+ excretion and congestion, which means it is like there should be some extra bodyguards, that there should be someone else who's, like in a lot of temples, they purposely, they forcibly push them out. So unless and until something like that happens, this will not be eliminated by natural mechanism. So let us assume how this can be achieved. So that's how you have a urinary buffer in the form of 2 buffers. One is called as a phosphate buffer. I can write in the luminal side. One is called a phosphate buffer, and second is called a ammonia buffer. Ammonia buffer. So I can probably write as ammonium also, or probably you can write as ammonia buffer. So these are 2 buffers are there. Remember, under physiological conditions, your phosphate buffer and ammonia buffer are equally important. Both can eliminate H+. But in times of acidosis, your phosphate buffer cannot expand, and it comes to a halt. It cannot expand its capacity. So it's not really important in times of acidosis, because there is too much amount of H+ that will be coming out of the distal nephron. So the ammonia buffer is the one that can be expanded in times of acidosis, and the phosphate buffer fails in times of acidosis, and it is something like a saturable, which means in our body, you can imagine that there is only fixed amount of phosphate reserve. We don't have that much amount of phosphate to increase the capacity, which means the phosphate buffer is something that is saturable, and you cannot expand beyond a limit. But ammonia is, as far as our body is concerned, ammonia is something that is infinite in capacity. You can get any amount of ammonia, even though infinity is it's an overstatement. Nothing can be infinite in this earth and world except for the universe itself. But nevertheless, considering the fact that our body has too much of ammonium, we can take that word for granted, that is having an infinite source of ammonia. So you can expand any amount of ammonia so that you can produce so much amount of elimination of acid. So how this ammonia buffer is going to be important. So that's the most interesting thing here. So let us assume, I told you one of the important functions of the proximal collimated tubule is to produce this ammonia. That's what we refer to as something called ammonia genesis. And where this ammonia is coming from, it is coming from the glutamine. So where this glutamine is coming from, it is coming from 2 areas. It is coming from the liver, and it is coming from the muscle as well. But the primary source under physiological conditions, even in our times of acidosis, is the liver. Liver is the principal source of glutamine production. And liver has two pathways. You know, if you take the liver per se, in the liver, this ammonia handling can be done by two areas. Where this ammonia is coming from, it is basically coming from the intestine by the action of intestinal bacteria. We know that in the intestine, the bacteria is the one that's going to produce ammonia. This ammonia load is handled by the liver basically. So this can be handled in two areas, one by producing glutamine and second by producing urea. So these are the two ways of handling ammonium by the urea. Urea is done by urea cycle and glutamine is created by glutamine cycle. So which means now you can understand where this glutamine is coming from. This glutamine is basically coming from the liver. That's what we need to know. So this glutamine using a lot of enzymes like PepCK is there, phosphoenol pyruvate carboxykinase, and we have a lot of other enzymes that is present in the kidney. They can convert this glutamine to produce ammonia. So this ammonia is something like a non-polar compound. So non-polar in the sense they are highly lipid soluble. I mean, there are some claims, there are some researches which are telling ammonia is not formed, they are forming basically ammonium. But I believe like both are same, because both are not going to make any difference. To keep it simple, let us assume they are producing ammonia. Now we can understand easily that this ammonia is a non-polar compound and it is going to freely diffuse into the interstitium. What I am writing now is the interstitium, not the cells. So in the interstitium, they are going to freely roam around because they are lipid soluble. They can move here and there easily. But actually this ammonium is actually struggling here because the pH is 7.4 in the body and ammonium's pK is 9.4. We know that. And this is actually literally struggling to become ammonium because at a pH of 7.4, according to Henderson-Hasselbalch equation, the natural tendency of ammonia is to exist in ammonium form. That's what we have studied some time back, not in the ammonia form. So this keeps on looking for H+, where the H+ is there, where the H+ is there, where the H+ is there. And of course, it enters the tubules because it's freely diffusible now. It enters the distal part of the nephron, keeps diffusing and waits to become ammonium. But at one point, it finds its partner. It is the H+ that is coming out in the distal nephron in large amounts. And now once it diffuses into the distal tubule, this ammonium and H+ will combine together to form NH4. That is ammonium. And that's what this ammonium wants. It's like a boy who searches for a girl all the time. And there are a lot of attractive girls that have been coming out of the distal nephron. And this single boy who's very frustrated and he wants to get committed and he sees that girl that is coming out of the door in the distal nephron, goes and hugs, and immediately it becomes committed and becomes NH4+. And as all the boys fade, once they become committed, once they become NH4+, they get trapped, which means they cannot move back. In pharmacology, one simple principle is once they become this ion forms, this protonated forms or any ionic forms cannot cross the cell membrane. One non-ionic forms can easily cross the cell membrane. So once they become ionic, they get trapped there. This is what we refer to as ammonial trap. And of course, this you can remember like Mangal Sutra, chloride also gets attached to that. And now their life is going to get eliminated in the urine, of course. And that's what we refer to as something called a ammonial trap. So this is one way of trapping the H+ and pushing it out in the urine, which means this ammonia buffer is very, very important to clear off the clear off the H+. As I told you here, initially, if you leave the people inside and automatically if you want to let them go out, they will not go out and they stay congested. And by natural movement, if they want to go out, you need a higher time and a lot of amount of urine. Suppose if you use a lot of bodyguards to push them out purposefully, forcibly, now they can move very fast. And of course, if you increase the number of bodyguards, you can increase more number of people that you can push out. Clear? So that's what is going to happen here. And this bodyguards are like buffer system, ammonium buffer. So you can imagine that this ammonium buffer can be imagined like that of bodyguards who forcibly push the H+ out of the system so that you clear these people all the time. You keep the auditorium clear so that more and more people can keeping on getting eliminated. So that is why if you take the graph that you see in a lot of textbooks, they are given a classical graph like this, where you will be seeing the buffer capacity and acidosis will increase in this particular area. And if you see the buffer capacity, buffer or how much they can buffer. So as H+ ions keep increasing in acidosis, the phosphate buffer will come to a halt. So this will be the phosphate buffer. But you take the ammonial buffer, ammonial buffer will keep on expanding. So it's basically infinite. So this is ammonia buffer. So ammonia is something that keep on expanding over a long period of time. So remember, it is the distal nephrons limitation to excrete only this much amount of H+. They cannot expand their capacity beyond this 700, or they cannot reduce the urinary pH below 4.5. So that's the least limit. So, but it's ammonia buffer that's going to be the most important. Glutamine to ammonia formation. Yes, to an extent. Small extent can happen in the distal nephron. Also glutamine to ammonia formation, but predominantly it is in the proximal nephron only. That is PCT only. I mean, this glutamine, if you really want to know how this forms, this is a basic biochemistry. I don't want to really complicate stuff here in this particular express session, because whenever I teach the detailed sessions, I take a lot of biochemistry and physiology also, but here you don't need that much biochemistry physiology, and I don't think really in the pattern what they're asking right now, you're going to get a lot of questions from those areas. Because what I felt in UPSC exam that held some 1 month back is like, there are so many questions where it is completely integrated into the, like medicine and surgery only. I feel like there have been like 110 to 120 questions on medicine alone in the UPSC paper. In the last year NEET also, if you take the overall number of questions, it's around 120 to 125 questions are from medicine and 70 to 80 questions are from surgery. The remaining small chunks they have been asking from different subjects. So it's really important to understand the effect rather than the basics more clearly. Basics are important, but if you at this point, especially if you're a post intern, you're digging so much of basics, then you're killing time and you're actually spoiling your chances. So try to avoid too much of basics and try to stick on to the amount of information that is relevant and which is needed to answer the questions, at least at this point of time. When you go to a post graduation or when you're an undergraduate 3rd year or 4th year, then that time you might be needing to dig in a lot. But when you're a post intern or an intern planning for an exam, this is not the right time to dig in with a lot of basics. Nevertheless, if you really want to know how glutamine is getting converted, this glutamine is will be initially converted to something called a glutamate. And this glutamate will be converted to something called a alpha ketoglutarate. And there are 2 enzymes that is important for this step. So the first step is the PDG, that is glutaminase enzyme. This is PDG, that's a glutaminase enzyme. Then we have another enzyme called the PepsiK. That is phosphoenol pyruvate carbose kinase. These are the 2 enzymes that is important, and at each step you will release an ammonia. At each step you will be releasing an ammonia. So this is the ammonia we are talking about. So in the kidney, this is the ammonia we are talking about that is going to, where is that? This is ammonia that is basically coming out here. I think you have rubbed it or not. Yeah, this is the ammonia we are talking about. So whatever I've drawn here, this is the ammonia we are basically talking about. And it's going to get pushed into the urine, of course. So this is what we refer to as ammonial trap. And there are 2 kinds of molecules that are coming out in the urinary buffer, in the urine as a buffer systems. Example, one is the ammonium chloride, and second is the H2PO4. This is dihydrogen phosphate, H2PO4, because we had another buffer also, that is hydrogen phosphate buffer. Where is that? We have another buffer, hydrogen phosphate buffer. This is the phosphate buffer we are talking about, and that is also coming out in the urine. That is dihydrogen phosphate. And this dihydrogen phosphate, we know the phosphate buffer's pK is approximately 6.8. That's the pK of the phosphate buffer. And as far as the pK of the ammonium buffer, we know it is 9.4. It is way above the this thing. So that is one of the reasons why ammonium buffer, because it's having a higher pK, it is supposed to be more effective buffer compared to that of the phosphate buffer. But this is something you can titrate basically. So once you titrate, for example, if you titrate to a pH of probably 7.4, that's what we do. This is what we call neutral titration. So this is the basic titration that we do. So once you titrate to a pH of 7.4, which means if you mix the phosphate buffer, that is H2PO4, to a pH of 7.4. So what will happen in this particular setting? But you can ask me, sir, how, if the pK is 7.8, that's what, I mean, pK is 6.8, you can ask me one important question. So many students do ask, if pK is 6.8 for HPO4 at a pH of 7.4, which means pK is less than pH. I mean, this phosphate buffer should be in dissociated form or undissociated form. At a pH of 7.4, this phosphate buffer will be in dissociated form or undissociated form. This phosphate will be in dissociated or undissociated form. Which means I can write like this, isn't it? HPO4 minus plus H plus is in equilibrium with H2PO4. So which means it will be in dissociated form or undissociated form. So at a pK of less than pH, so it should be in the dissociated form. That's what all your understanding is. So many students do ask me a question, sir, then how come you can form this H2PO4? Which means the whole concept of buffer itself is getting lost, isn't it? This H plus, if it doesn't mix with HPO4 in the first place at a pH of 7.4, the whole concept of buffer itself is lost completely. You know, like you cannot produce H2PO4 in the sense how this H plus can be washed out by HPO4 if it doesn't combine at a pH of 7.4. So answer is very simple. That is what this H plus is doing here. Basically, this H plus coming out into the urine will actually lower the pH of the urine. So which means now the pH of the urine is what you're talking about, not the neutral pH. In the urine pH, I told you it will be say 4.5 to 6.5. This will be the urinary pH. And why the urine is acidic? Because of the movement of the H plus into the urine. And they're going to reduce the pH. Now you can assume that the more the urine is getting acidic. So this is like kind of a very beautiful example. The more the pH is less. So for example, at a pH of 6, if you take the pH of pK of the phosphate buffer is 6.8, at a lower pH, definitely they tend to form more H2PO4, which means more H plus can be trapped. When it becomes 5, you can produce more and more H2PO4, more and more H2PO4, and you can trap more HPO4. So that is the beauty here. So which means the urinary pH is acidic for that reason only, to trap this H plus, and vice versa. Both this help each other. So they trap H plus and they tend to take this H plus off. As they take the H plus off, depending on the incoming H plus, depending on the lowering of the urinary pH, more and more H2PO4 will be formed. So what we are trying to tell is, it is not the pK that is important here, it's the urinary pH that is important. Usually it will be 4.5 to 6.5. That's the urinary pH, which is actually in this situation, it is less than that. I mean, pK here is more than that of the urinary pH. So H2PO4 will be existing in undissociated H2PO4 form only, so that they are trapping more and more H plus, resulting in an active state of a buffer like a scenario in the urine. So that's what you need to understand. And at the same time, let us suppose urinary pH is 4.5 to 6.5, and let us assume a patient who's having a urinary pH of 5.5, a normal patient like me or you who's having urinary pH of 5.5. Now I know, because in this situation, my pKa is more than that of the pH, which means this urine will contain a lot of H2PO4, because here the pK is more than pH and they will be in undissociated form. So they have trapped some amount of H plus and they are leaking into the urine outside the body. So now I need to know how much amount of H plus is trapped by this H2PO4. So how much amount of H plus is actually trapped, or how much amount of H plus I can I'll be seeing in the setting of H2PO4. So for that, what I need to know is very simple. I'll take this urine and titrate. I'll take this urine and I will titrate with the solution outside in the body. With the solution, I will titrate with the pH of 7.4, which is the neutral pH. That is 7.4. So once I titrate this H2PO4, which is having a pK of 6.8, and I'm now titrating with a solution that is containing a pH of 7.4. And slowly what happens, because now the situation is reversed now the pH is more than pK, or pK is less than pH. Now the H2PO4 cannot remain in H2PO4 form. So now slowly they will start releasing more and more H plus into the solution. So which means when I titrate, my H plus will keep on releasing. And I can exactly find out at the neutralizing point, at the end point that is achieving a pH of 7.4, at that point, I know exactly how much amount of H plus that is present in the H2PO4 form. So that is why this is also referred to as something called a titrable acidity. You would have all done in this kind of experiment in your 11th standard or 12th standard chemistry, where you do that phenolphthalein test where you achieve a neutral color. That white color, when it comes, that's the point of titration. You know how much is the H plus ions that is getting that is present in a particular solution. So this is what we refer to as something called a titrable acidity or a titratable acidity, which means by titrating with a pH of 7.4, you know how much amount of H plus is approximately trapped by H2PO4, which means you can know, you can titrate it. That's why it's called a titratable acidity. So in exam, if they ask you what do you mean by titratable acidity, very simple. It is the amount of H plus that is trapped in the H2PO4 buffer. That's a phosphate buffer. That's what we refer to as a titratable acidity. On the other hand, the ammonia cannot be titrated, and it's a well-known fact because its pK is 9.4. Even if, I mean, that is also one reason why it is a very effective buffer. Because suppose if the urine pH becomes alkaline, for example, if urine pH is 7 for that matters, your phosphate buffer will be gone. You know, like because the pK of the phosphate buffer is 6.8 and you cannot trap any more H plus at all. That's all gone. You cannot do anything. That's why physiologically this is very important. But ammonia buffer is effective at all states because even if the urinary pH becomes 7, your pK of ammonia buffer is 9.4, which means still the relationship is the fact that here the pH is less than that of the pK. Here relationship is still the pH is less than that of the pK. Still they'll be able to trap more and more H plus and form more and more ammonium. That's why ammonia buffer is extremely effective buffer in all the situations in the body. I don't think urinary pH will reach 9.4 at any stage in your life. Even if you're dead, it's not going to reach 9.4. But that's why I'm telling you, ammonia buffer is extremely important buffer as far as kidney is concerned because it can keep eliminating H plus even at any pH of the urine. Doesn't matter. So it's a very, very extremely effective buffer, unlike your phosphate buffer, which is having a pK of 6.8, and it will definitely fail once the urinary pH becomes more than 6.8 or anywhere near 7. But if ammonia buffer, since it's at 7, they can effectively trap H plus, and even at 5, they will trap more H plus. At pH of 4.5, they are going to trap extremely high amounts of H plus. So more the difference between pK and pH, more they are going to trap H plus and they're going to exist in ammonium form because of this pK-pH relationship. That is why the understanding of the intranasal bulk rules becomes a very important thing, especially when you wanted to understand this kind of basic acid-base physiology. But what I was trying to tell is this ammonium chloride cannot be titrated because the pH is 9.4. If you keep a solution pH 7.4 that is neutral or solution pH of 7, I don't need this. Why I need this really? Why I want to keep the pH 7.4? Even if I keep a pH of 7.4, even in that scenario, my pH will be less than that of pK as far as the ammonium buffer is concerned, and they are not going to release H plus at all in the first place. And titration at a neutral body's pH has nothing to do with the ammonium buffer. So that is why this is not titratable. You cannot titrate. This is not referred to as something called a titratable acidity. Whereas phosphate buffer, you can titrate. That's why it's called a titratable acidity. On the other hand, one more important thing is, but this ammonium is something that can be measured directly. So that's why there is a measurement called urinary ammonium. So you can actually measure the urinary ammonium basically, because if you can measure the chloride, that chloride will be to an extent surrogate marker of how much ammonium is released as well. Because ammonium anyway will be coming in ammonium chloride form, only it will not be coming in pure ammonium. So the chloride measurement to an extent can give a surrogate marker of how much ammonium is getting excreted. At the same time, there are separate assays where you can find out how much ammonium is there. But most of the time, we don't use this kind of cumbersome measurements and all. Just chloride analysis itself is more than enough, which can approximately tell how much ammonium is coming out. So now you need to know that that phosphate is the titratable acidity, that ammonium is not basically a titratable acidity. So now you can understand how this distilled nephron is very important for eliminating the fixed acids that are produced in day-to-day life. And at the same time, if you take your beta intercalated cell. So beta intercalated cell is important times of alkalosis. I told you. So it's important times of alkalosis. How these cells are important times of alkalosis, what you're going to see. Remember, it's the same transporter. We are seeing the bicarbonate chloride exchanger here that we have seen. It's the same exchanger, but you just reverse the polarity and change the position. For example, in a beta intercalated cell, your chloride bicarbonate exchanger will be located in the apical membrane, whereas an alpha intercalated cell is located in the basolateral membrane. Whereas in beta intercalated cell, just you have changed the polarity in a reverse direction, and just you have kept it in the apical side instead of the basolateral side. Now this particular transporter now will be able to eliminate bicarbonate and they will reabsorb chloride. And this is a very important situation because in times of alkalosis, you are going to have more bicarbonate. We know that. So to eliminate bicarbonate, this transporter becomes really, really important in times of alkalosis. It's not important through all the times, but in times of alkalosis, this becomes really important. And this is also important understanding one important principle that if you are giving too much of sodium chloride solution, too much of sodium chloride solution, it will result in acidosis or alkalosis. Simple question. It will result in acidosis or alkalosis. Too much sodium chloride, which means I'm talking about a normal saline. Too much normal saline will result in acidosis or alkalosis. Or it will be a neutral, no change in the pH. So third option, no change in the pH. Acidosis, alkalosis, no change in the pH. So what will happen if you too much of sodium chloride? The one answer of acidosis and two answer of acidosis, three answer of acidosis, two answer of alkalosis, three answer of acidosis, three alkalosis, four acidosis, four alkalosis. No, okay. So it's a 50-50. Some are telling acidosis, some are telling alkalosis. But it's a simple thing. It's called acidosis. That's called a hyperchloremic acidosis. Why hyperchloremia results in acidosis? Whenever you are going to induce a kind of hyperchloremia in the body, basically if you are producing more chloride in the body, which means the serum contains more chloride. If you are producing hyperchloremia, of course, the glomerular filtrate will also contain more chloride because whatever is in the serum only that's going to be transformed as glomerular filtrate. So glomerular filtrate also will contain more chloride. If the filtrate and the urine contains more chloride, this more chloride will act as a stimulus for this transporter to excrete more bicarbonate. If you excrete more bicarbonate, you will result in a situation of low serum bicarbonate in the setting of high chloride scenario. So this is what we refer to as a hyperchloremic metabolic acidosis. That's very important. So too much sodium chloride can itself induce a sort of acidosis, but that is completely reversible in a short span of time. Once the acidosis goes off, this will completely go off. That's called as hyperchloremic metabolic acidosis. Especially you can see this kind of hyperchloremic acidosis in the setting of trauma scenario or decay scenario, where you give too much of sodium chloride containing solutions. Even RL can produce this kind of a scenario, but because RL is more balanced and they will not result in hyperchloremic acidosis because RL also it contains sodium chloride, but it will contain something called lactate as well. This lactate will go to the liver and they will get regenerated as bicarbonate. So because this lactate gets regenerated as bicarbonate in the liver, so that's a different sort of a scenario. So RL will have lesser effect on hyperchloremic metabolic acidosis compared to the original sodium chloride solution. Sodium chloride can produce more incidence of hyperchloremic metabolic acidosis if you are giving too much of sodium chloride solutions. That's one point that you need to know for sure. I think now the physiology must be very clear. So, I mean, this is the maximum physiology I'm going to discuss, and this must be clear for you. Do you understand? So that we can move on to the next one. So the actual acid-base problems. Okay. So once you have understood this basic physiology, it is important to understand the acid-base issues also that is happening in the body. So even though the Henderson-Hasselbalch equation will help you in understanding almost all the types of acid-base disorders, but after that, there are two nephrologists who have told the problems extremely easily, and in a very simple equation, they have told the problems as far as the acidosis and alkalosis is concerned. So what equation they have given is that pH of the body is proportional to the bicarbonate divided by carbon dioxide concentration. This is what they give. So this is a much simplified equation, or alternatively you can write, because pH is inversely proportional to H+. pH is inversely proportional to H+ concentration, which means more the H+, lesser the pH, acidosis. That's what we talk about. So or you can write that H+ concentration is is proportional to CO2 by HCO3. Both are correct. Both are absolutely correct. Here, what I have done is I have just inverted the equation, nothing else. I have just changed the equation, just have inverted the equation. That's all. So because H+ is inverse of pH, we know that. So that's why I inverted the equation. So what they have told is in a very simplified way for students to understand, especially for not for complicated math, but for students to understand, pH you can write as it's proportional to bicarbonate by carbon dioxide. This is very simple. Suppose if you increase the carbon dioxide by any reason, of course the pH should reduce because we know carbon dioxide is a volatile acid. So pH should reduce. Suppose if you are going to increase the bicarbonate, the pH should increase because it's directly proportional, because we know bicarbonate is alkali and increase in bicarbonate should result in alkalosis only. So based on this equation, we can give a very simplified table. So what happens in the setting of metabolic acidosis? What happens in the setting of metabolic alkalosis? What happens in the setting of respiratory acidosis? And what happens in the setting of respiratory alkalosis? First, let us see in metabolic acidosis what will happen. The first scenario is pH is proportional to, we know that bicarbonate and carbon dioxide. This is the same formula I want to write for everything to understand. So first, let us see the primary problem here. Primary issue in metabolic acidosis is definitely going to be decrease in the bicarbonate, which means you are trying to increase the H+. That increase H+ effect is going to be seen in the body as reduced bicarbonate, because H+ when it is increasing, of course, it is going to mix with the bicarbonate. So that the bicarbonate reserve will be lost. So bicarbonate amount will be shown as low only. That will result in reduced amount of, because reserve will be there, but more H+ will trap more bicarbonate, resulting in more H2CO3 production. This more H2CO3 production by carbonic anhydrase mechanism, they will result in more CO2 formation, and this CO2 can be eliminated. So that is the idea. So which means you are basically trapping this H+ to produce carbonic acid to produce more CO2, which can be eliminated. And that is how, your Henderson-Hasselbalch equation works. And that's what happens in the setting of acidosis also. So more H+, you eliminate more CO2. So because this H+ is what is eliminated in the form of CO2, which is actually carrying that proton, that is H+ in it. All right. Now you can understand this is in a different way also. Like for example, body produces a compensatory response. How the body produces? Because the body now knows that more H+ is getting generated somehow. Because more H+ is getting generated, now the body tries to eliminate more and more CO2. It's like pulling the reaction towards that particular side. So now what they're trying to do is they're eliminating more CO2, means if they eliminate more CO2, now they can accelerate this reaction because it's like sucking with the negative pressure to that particular side, because you're eliminating more and more CO2, and which means you'll be producing more and more carbonic acid. To produce more and more carbonic acid, this reaction should happen in an accelerated fashion. This H+ also will be trapped more and more, which means you can rapidly down-regulate the H+ and eliminate out of the system. So which means the body does a compensatory response by doing something called hyperventilation. If they do hyperventilation, they can eliminate more CO2. Body helps by doing hyperventilation. There are two responses for this. One reason to eliminate more CO2 is because in times of increased H+, that is acidosis, your pH will drop. And how can you, increase the pH back? To increase the pH back to normal, if you want to bring the pH back to neutral range, you need to somehow reduce the H+. This reduction the H+ can be done by eliminating carbon dioxide. So because this contains that proton which I was talking about, this carbon dioxide. So once you eliminate more and more, you can reduce the H+ more and more. And at the same time, this also accelerates the forward reaction. Accelerated reactions will happen, which means this H+ can be trapped more and more, and this forward reaction will proceed faster so that more CO2 getting eliminated, so more H+ can be cleared. So which means one of the important body's response to your metabolic acidosis is hyperventilation response. So that is why your bicarbonate is low, means your H+ is eventually high. So this is a sign of an acidosis, metabolic acidosis, and the response of the body to this metabolic acidosis increased carbon dioxide elimination. That is hyperventilation. This is so clear and so cool. And you can explain everything based on this Henderson-Hasselbalch equation, but it will be too tough and too time consuming and unnecessary for you right now. Anyway, you're going to forget, and after sleeping tonight and waking up tomorrow morning, you will not be even knowing what I have told in the last 2 hours. So that's how Anderson-Hasselbalch bulk equation works. So let us keep it simple. We don't want the Anderson-Hasselbalch bulk equation. The primary issue is increase H+, the fixed assets are increasing. That's why it's called metabolic acidosis. This will be seen as low bicarbonate in the body. The bicarbonate reserve will be low. That's why the bicarbonate levels are low. So this is the evidence of the metabolic acidosis that is ongoing. So what will be the basic compensatory response here? So compensatory response, let me write in green. So now you know. So if your acids are increasing in the body, you have to eliminate the more acids. But because this is a fixed acid that is getting increased and the problem is in the metabolic component, the body tries to eliminate more and more volatile acids. So if they eliminate more and more volatile acids, then only your pH can be maintained in the normal range. So which means you can understand that. So now the HCO3 is decreasing, which means pH is going to drop, but I don't want that drop to happen. So I want to maintain the pH in the normal range. To relatively increase the pH now, what should be the compensatory response? I need to reduce CO2 or I need to increase CO2 to maintain pH in the neutral range. So it's your take. So see the equation and you can tell. Because HCO3 reduction is reducing the pH towards acidotic range. And to maintain the pH in the normal range, you need to reduce CO2 or you need to increase CO2. So it's your take. People have to tell. To maintain this equation, it's a simplified one. As simple as that, isn't it? You need to actually reduce the CO2 to maintain the pH in the normal range. So which means this is going to be the compensatory response to a metabolic acidosis. So as a compensatory response, my CO2 levels are decreasing, my carbon dioxide levels are decreasing in the body, and this is an absolute compensatory response. So now you take a metabolic alkalosis. Metabolic alkalosis, let us see what happens in the setting of metabolic alkalosis. Metabolic alkalosis, same equation, pH is proportional to bicarbonate by carbon dioxide. Same, absolutely same, I'm not changing anything. Here the primary issue is very simple. If I talk about metabolic alkalosis, which means my pH is increasing because of increase in here the primary problem is reduced pH. Here the primary problem is increased pH, alkalotic pH because bicarbonate is increasing. And that primary problem issue here is the reduced H+, that is actually resulting in increased bicarbonate, which is a surrogate marker of H+ in real time. So this is what we refer to as alkalosis. And more importantly, you think that bicarbonate is acting like a alkali or acid. No, bicarbonate is only a marker of acidosis or alkalosis. It's per se thinking bicarbonate is an alkali is wrong. But nevertheless, we're not talking about pure chemistry and we're not going to be any chemistry PhD. We're not going to get like that. So it's okay to tell that bicarbonate is an alkali for our understanding. But actually speaking, bicarbonate is not an alkali. It's only a marker of underlying acidosis or alkalosis that is going on. So low H+ is the alkalosis that is happening here, and that is manifesting as more bicarbonate according to the Anderson-Hasselbalch bulk equation. And now as far as the compensated response, now you are going to answer. It's quite simple. The carbon dioxide should increase or decrease. Should increase or decrease to bring the pH back to the normal range, to the neutral range. What should happen to the carbon dioxide here? It should increase or it should decrease to neutralize that pH change, because the compensatory change is happening to neutralize the change in the pH. So to prevent the rise in the pH here, the carbon dioxide should increase or decrease. It should, of course, increase, and the compensatory change is going to be the change in CO2. That is increased CO2. Here to reduce CO2, I need to produce I should not call it respiratory alkalosis, I should call hyperventilation. That is a compensatory response here. Similarly, to increase the CO2, I need to produce a hypoventilation. So that's the compensatory response in response to metabolic alkalosis. Then you take respiratory alkalosis or respiratory acidosis for that matter. Here the primary issue I know. Here the primary issue, if I tell respiratory acidosis, the primary issue must be hypoventilation, because I'm not, remember, hypoventilation acting as a compensatory response and hypoventilation happening due to a head injury. Because of that, I'm not breathing properly, something completely different. So here the primary issue is hypoventilation, not the compensatory problem. The primary issue is hypoventilation, that is increasing the carbon dioxide. If I'm increasing the carbon dioxide, of course my H+ will increase. Why? By Henderson-Hasselbalch bulk equation, H+ plus HCO3 minus is H2CO3, which will result in H2O and CO2, and everything is completely reversible. We know that. So now, due to some reason, I'm not eliminating this CO2 because my way of elimination of CO2 is lost and I'm accumulating CO2. If I'm going to accumulate CO2, the entire reaction will be affected. I cannot produce H2CO because H2CO3 cannot move to the other side. If H2CO3 cannot move to the other side, this H plus and HCO3 minus also cannot become H2CO3, so that my H plus will increase in the body. If there is only one way to push this reaction forward. You all know that. So that is the reason why we call this as acidosis, because you can imagine like my way of eliminating this proton, volatile acid is lost here. So that's why my proton in the body is increasing. That's what is causing acidosis. Because of hyperventilation, that's called respiratory acidosis, because the respiratory component here. As a compensation, the only way I can eliminate these protons, I can reduce the amount of protons, is only one way. If you have a normal bicarbonate, now they cannot handle this much amount of increased H plus. So my only way is to conserve H plus and to increase the conserve bicarbonate and increase the bicarbonate. Then only somehow I can push this reaction forward, and somehow I can eliminate some amount of H plus that is getting accumulated because of the poor ability of the body to eliminate protons in this scenario. So that is why the compensatory response is to conserve bicarbonate and to increase the body's bicarbonate reserve. That's what happens in the setting of a respiratory acidosis. But everything you can explain with the Henderson-Hasselbalch equation itself, but that's not important. Again, as I told you, you all need is this reaction to be understood properly. Bicarbonate by CO2. Suppose if your pH is going to, because here the primary problem is carbon dioxide is increasing. Because of that, my pH is decreasing. That's a primary issue here, hypoventilation. Suppose if I should have a compensated response here in the metabolic side, if I should have a compensated response, what should happen to the bicarbonate? The bicarbonate should increase or bicarbonate should decrease to bring the pH down to normal, which means to increase the pH back to normal. Bicarbonate should increase or bicarbonate should decrease to bring the pH back to normal, which means here the problem is pH has already reduced because of high carbon dioxide. So the pH here the bicarbonate has to, yes, as all you told, the bicarbonate has to increase to bring the pH back to the normal range. To increase the pH and to bring the pH back to normal range, the bicarbonate should actually increase. That's what happens here. Here the bicarbonate values will increase, and this is done by the kidneys. And this is what we refer to as a kidney compensation or metabolic compensation or conserving bicarbonate. That's what happens. That's what the kidneys does by understanding this entity. And number 4, you have something called a respiratory alkalosis. Here, you know the pH, same equation. This is the only master equation that you need to know. This is the same equation that will be useful. Here, my primary issue in respiratory alkalosis, because of some reason I am hyperventilating. Because of hyperventilation, I am washing out more and more CO2, and this could be a salicylate poisoning. Something is there, but don't think about that. It's a respiratory alkalosis because of some reason I'm anxious, hyperventilating voluntarily, and because of that, my CO2 is getting washed out. Because CO2 is decreasing, my pH is increasing, going towards alkalotic range. Because more CO2 loss, more proton loss, alkalosis. That's what is happening here. So in this situation, what should be the compensatory issue? It's very simple. Now, to bring this increased pH down to the normal range, you should reduce the bicarbonate or you should increase the bicarbonate. So what should happen? Decrease or increase? What should happen to the bicarbonate here? Of course, everyone knows that you need to reduce the bicarbonate in this particular setting. So once I reduce the bicarbonate, that will act as a compensatory response to bring this pH back to the normal range, which means here the kidneys try to eliminate more bicarbonate, which means they try to excrete more bicarbonate. So the kidneys eliminate bicarbonate as a compensatory response. So whatever I have drawn here in red are the actual primary problem, and whatever I have drawn in green in this particular examples are actually compensatory responses. So there is one point that you're noting here. There are 2-3 takeaway from here. One point that you clearly note here is the fact that the arrows are basically moving in the same direction. If you see everywhere in all the equations I have drawn, if you see bicarbonate and carbon dioxide, if you take their values, the arrows literally move in the same direction. They never move in the opposite direction. They are going to move in the same direction only if it's an uncomplicated single acid-based problem. If the problem is either in the metabolic side or if the problem is in the respiratory side, not both. That's called a single uncomplicated acid-base disorder. Problem is in the respiratory side alone, or the problem is the metabolic side alone, then definitely you should see that the arrows should move in the same direction only. It can never move in the opposite direction. It is not possible. For example, if I am actually having a metabolic acidosis, my body's response should be to eliminate more protons from the system through the volatile acid, that is carbon dioxide. I cannot keep conserving more and more carbon dioxide, and unless and until there is a problem in the respiratory component also, where I'm getting a head injury and not breathing properly. So unless and until we get that kind of scenario, you're not going to result in increase in the CO2. If there is a metabolic acidosis, low HCO3 means there should be a low CO2 only, and then only you can call it as a compensatory response in the first place. So to call it as a compensatory response, and that is acting fair and good, your compensatory response should always move in the same direction. It means the arrow should move in the same direction as that of your metabolic component. It means bicarbonate decrease, CO2 decrease. Bicarbonate increase, CO2 increase, or CO2 increase, bicarbonate should increase, and CO2 decrease means bicarbonate should decrease. Any change, any deviation from this. For example, if I'm seeing that arrows are moving in the opposite direction, one is moving in one place, another is moving another place, or one is decreasing, another is increasing. So if you see this kind of opposite direction arrows in the equation, that is 100% suggestive of a mixed problem. It cannot be a single acid-base disorder. It must be a mixed problem. It's not uncomplicated anymore. It is a complicated acid-base disorder, which means I'm having problems in both metabolic area also. I'm having problem in the respiratory area also. Arrows cannot move in the opposite direction. That's the first take-home point from this, whatever I have told you till now. Let me ask you a second question. Suppose if the pH is like this, let us assume here the problem is the metabolic acidosis, which means now the bicarbonate has decreased because the pH has decreased and has become 7.2. We know the normal pH is 7.4. Now it has become 7.2. Now the carbon dioxide is decreasing, and why the carbon dioxide decreasing? Can anyone answer why the carbon dioxide is decreasing here? I mean, you know it's a compensatory response, but why the compensation is happening? That's what I'm asking. Why the CO2 is decreasing? According to this equation itself, you can tell what is the reason for decrease in the CO2. It's a very easy question I'm asking. Yeah, Keshavan told the right answer, but I'm expecting answers from many other people also, because so many people is there. So many people are there. Why the carbon dioxide is decreasing here? The carbon dioxide is decreasing, of course, yes. One person is telling that to increase pH, to eliminate acid. But what is the primary reason for this compensation to happen? That is to maintain the pH. Why they want to do that? Because they want to maintain the pH. Okay, fine. Now let us assume a second scenario. pH is there. I mean, now I told you the first scenario where the reason is to maintain the pH, to bring the pH back to normal. That's why the carbon dioxide is reducing. So pH is proportional to bicarbonate by carbon dioxide. Now assume a primary respiratory acidosis. Now there is a respiratory acidosis because the pH is dropping. Now if my bicarbonate levels is increasing as a compensation, now you know why this bicarbonate is increasing as compensation. This is because my pH has to increase back and I have to bring the pH back to normal. So now let us assume my pH has come back to normal. Now my pH has become, now it was initially, it was 7.2. Now it has become 7.35. My pH has become normal almost. So now the equation stands like this. pH is 7.35. Bicarbonate has increased and CO2 has increased. So here CO2 is the primary issue. Bicarbonate is a compensatory problem. So if you listen carefully, if you want me to tell one more thing, my question is that can bicarbonate increase further? I mean, can it keep on increasing further because CO2 is increased? Can it keep on increasing further to make the pH 7.5? To make the pH 7.5, is it possible? Is it possible for a compensatory response to increase the pH to 7.5 in this particular example? Yes or no? I want only yes or no answer. Will this be possible? Can the pH become 7.5 just because the compensatory response? Yes or no? Dr. D is telling yes. Actually, answer is no. You can't do that because I told you very clearly the role of the compensatory response. The role of the compensatory response is to maintain the pH in the normal range. That's all. So not to create another acid-base problem. So that is why in your biochemistry textbooks, they would have given one more point, that there is no compensatory response that can overcompensate, in the sense like they can either undercompensate or they can fully compensate. That's called a partial compensation or a full compensation. There is nothing like overcompensation in the body. Your compensatory response cannot overcompensate, and they will increase the pH or change the pH in the reverse direction, which means the role of the compensatory response is to bring the pH back to normal. So you cannot expect the compensatory response to create another acid-base problem, creating another direction you bring the pH, and that's nonsense. And that will not happen. That's why it should be extremely clear that the compensatory response is going to try and bring the pH back to normal. It may succeed or may not succeed, but it can never bring the pH to that other end extreme direction. Not possible. So overcompensation is not possible in our body. It's always either under partial compensation or probably maybe a full compensation. If the pH has come back to normal, it's full compensation. The pH has still not come back to normal, but still the compensation working, means it's a partial compensation. So body can never overcompensate. That's a very important point to note. And next point that you should notice, one important problem here. Another point is that for a metabolic problem, your compensation is respiratory. We know. I mean, you can like ask me like, sir, we know that. So, but let me tell you one more point here. So, a metabolic problem means the compensation is a respiratory compensation. So, what I'm trying to tell is, in a metabolic problem, in a primary metabolic issue, your compensation is respiratory compensation, and this respiratory compensation is going to be fast, very, very fast. In the sense like it will happen so fast. But even though if it is fast, but it cannot be complete. It's usually only a partial compensation. It won't be effective compensation. It will be only a partial compensation, which means this change in the carbon dioxide that is happening because of a primary metabolic issue, the respiratory component, that is carbon dioxide, will change immediately, which means it's fast. Fast means I'm talking about immediate changes. It will change very fast, but will not be able to completely compensate, which means it will be only always a partial compensation. It will not be a complete compensation, which means it will not be able to bring the pH back to normal. For example, the pH is 7.2, bicarbonate is reduced, acidosis, PCO2 is also reducing as a compensation, means it can bring the pH probably to 7.29, but it will not be able to make 7.4, not possible. This PCO2 drop can correct the pH to an extent, but it cannot bring the pH back to normal. That is how it happens, because the respiratory compensations are basically weak compensations. So they cannot bring the pH back to normal. So which means it will always be a under compensation or probably a partial compensation only, but it's extremely fast. It acts rapidly because you're breathing every second, every minute you breathe. So it acts very fast, very rapidly. So fast, but not very effective. On the other hand, for a primary respiratory problem, so if you think there's a primary respiratory problem, your compensation is metabolic compensation. For a primary respiratory issue, your compensation is a metabolic compensation. So metabolic compensations will take usually 48 to 72 hours, which means it's done by the kidneys. So it will take some time. So it's a little late. It will not start immediately within seconds. It will take some nice 48, 72 hours to kick on. But even if it comes late, it will be effective. Effective means it usually, most of the times, metabolic compensations will be full compensation, especially for chronic respiratory disorders. Metabolic compensations will be effective and usually will be a full compensation most of the times. Kidneys are very effective in actually compensating for the acid-base disorders. So respiratory compensations fast, but not that effective. Metabolic compensations slow, but extremely effective, especially for chronic respiratory conditions. Slow means only for chronic it can work. Acute respiratory scenarios, it won't work. So that's what the derivations we are going to make. So for example, if you have a metabolic disorder, the compensation is going to be respiratory compensation. So there'll be respiratory response, compensatory respiratory response. This respiratory compensation tends to be fast, but very partial only. Fine. So similarly, if you take a respiratory disorder, it will be late. The compensation will be metabolic compensation. Compensation will be a metabolic compensation. It will be late, but effective, done by the kidneys. So I can derive two here. Because it is late, the first point I'm telling, because it is late, in an acute respiratory disorder, there'll be no compensation. which means they will be uncompensated. There will be absolutely no compensation because they come very late. So in an acute respiratory disorder that's happening within hours, 1 or 2 hours, there will be absolutely no change in the metabolic component, and there will be absolutely no compensation. But as well as in a chronic respiratory disorder, if the patient is suffering from a chronic respiratory issue, usually there will be a full compensation most of the time. This is very, very important. There are 3 gold standard points I've told you. In a metabolic disorder, usually you experience a partial compensation. In a respiratory disorder, it depends on acute or chronic, like an all or none phenomenon. In acute respiratory disorder, because it's very late to kick in, there will be no compensation. But once it comes, it becomes effective. So in a chronic respiratory disorder, there will be a full compensation. So in exam, this partial, uncompensated, full compensated. So don't get confused. Whenever you have metabolic disorder, it's very likely to be a partial compensation. Whenever you have a respiratory disorder, see whether it's acute or chronic. Acute respiratory disorder means usually it will be uncompensated. Chronic respiratory disorder means usually it will be completely compensated or fully compensated. So that's the take home point here. Okay. Now having understood this important thing, so let us put the basic rules for ABG. Basic rules in an ABG. So first we are talking about an uncomplicated single acid based. I mean, if it's an uncomplicated single acid based disorder, we know that arrows should move in the same direction. Whether it's up or down doesn't matter. Arrows should move in the same direction. Rule number 1, if the arrows are moving in the opposite direction, definitely it's a mixed disorder. It's not an uncomplicated disorder anymore. It's definitely a complicated and a mixed acid based disorder. Point number 1, arrows should never move in the opposite direction. Very, very important point. Let us assume now you have a primary metabolic disorder. In a primary metabolic disorder, what is the problem here? Here there is a change in the bicarbonate. That is the primary issue here. And as a compensated response, there should be a change in the carbon dioxide. This is a compensated response. And this change in the carbon dioxide should happen very fast in metabolic disorder because respiratory compensations are too fast. We know that. But I am telling, I'm not seeing any change in the carbon dioxide. I'm seeing a normal carbon dioxide. What does that mean? So what you will tell here? What is the problem here? So it's a primary metabolic disorder. I'm changing a tremendous change in the bicarbonate, but my carbon dioxide is failing to move on either side. So what is the inference here? What is the inference? See here, inference is not no respiratory compensation. I told you all, that's why I'm telling there is no respiratory compensation. So respiratory compensation should happen immediately. But if there is no respiratory compensation, what is the inference? This indicates the patient is having a mixed acid-base disorder again. I mean, I'm not asking about a brainstem problem now. See, Lakshmipriya is telling brainstem problem. I mean, it could be anything. So, for example, if you have a failure of respiratory compensation, I can tell anything, right? From brainstem, or cortical problem will not cause this. Neuromuscular issues will not cause this or what. I can tell, I'm not asking the cause. I'm asking about whether it's mixed or single. It is a mixed disorder, of course, because my respiratory part is supposed to move in a metabolic disorder, and it has to be fast. It has to move within seconds. It has to move. If it is not moving, then that should be definitely a mixed disorder. Very, very important. So in a primary metabolic disorder, if you see a normal carbon dioxide, it always indicates a mixed disorder. Then, I mean, number 3, I'm asking a primary respiratory disorder. Now it's a respiratory event. We know here the primary problem is change in the carbon dioxide, and that's the primary problem. If this is the primary problem, I'm telling now I'm having a normal bicarbonate. What is the inference here? In a primary respiratory issue, if I'm having a normal bicarbonate, what is the issue here? What is the problem? Is it a mixed or is it a single disorder? No, it's not a mixed disorder. I mean, to start as a beginner, if you have a normal bicarbonate in this scenario, so there is only one possibility usually. I mean, if you don't have any other logic, the one logic is it must be an acute respiratory disorder. So we know that in acute respiratory disorder, the compensation will not happen and it's uncompensated. So in an acute respiratory disorder, this is what is expected. That's a quite normal thing to see. Bicarbonate will not change that much. Similarly, if I have a changed bicarbonate, a significant change in the bicarbonate, but in the same direction, we are talking about opposite means it always goes towards a mixed disorder. Whenever there is a significant change in the bicarbonate, I can call it as a chronic respiratory disorder. So which means in the acute respiratory disorder, the bicarbonate is expected to be normal. And in a chronic respiratory disorder, the bicarbonate is expected to change, but of course in the same direction and appropriately, not in the opposite. Opposite means it will become a mixed disorder. Now rule number 4. Now I'm talking about a primary respiratory disorder. Again, I'm clearly giving it's an acute primary respiratory disorder. Now you know the carbon dioxide change will be the primary issue here. The PCO2 is going to change and the carbon dioxide change is going to be the primary issue. If this is going to be the primary issue, what is going, I mean, if I should actually expect a normal bicarbonate, because in an acute respiratory event, as I told you, there should be a normal bicarbonate only. Suppose if I'm changing a grossly changed bicarbonate. So what is the inference here? Is this correct or is this incorrect? I'm talking about an acute respiratory disorder and a grossly changed bicarbonate. Is it correct or incorrect, or it's single disorder or mixed disorder? Of course, as you all told, it's correct. It's supposed to be a definitely mixed disorder. And this is not correct. This is not something that is expected from an acute respiratory disorder. Similarly, if you have a primary respiratory disorder that is chronic in nature, I'm talking about a chronic respiratory disorder. Of course, it will be a change in the CO2 that will be a primary event here. And in a chronic respiratory disorder, I'm actually expecting a sort of changed bicarbonate, but I'm not seeing a changed bicarbonate. Instead, I'm seeing a normal bicarbonate, not a changed bicarbonate. Instead, I'm seeing a normal bicarbonate. Normal bicarbonate. What does that mean? If you have a normal bicarbonate, what does that tell? It's it's okay or is it not okay? It's correct or incorrect. So you're talking about a chronic respiratory disorder. It's clear. And I'm having a normal bicarbonate. Is it correct or incorrect? So what does that mean? Is it correct or is it incorrect? Definitely it's correct. It should not happen because we expect the bicarbonate to change dramatically, but of course in the same direction. We're talking about opposite direction, in the same direction. So it's again going to be a mixed disorder. Clear, very, very simple. So ABG is something you'd expect and it doesn't, it doesn't fulfill your expectation. You call it as a mixed disorder. That's all. So whenever I expect something in ABG to happen, and if it doesn't happen, it goes against your expectation, then it's a mixed disorder. That's how. If it doesn't satisfy you, it doesn't get correctly as per your expectation, it's called a mixed disorder. So in a single acid-base uncomplicated disorder, we don't see opposite arrows. And of course, any opposite arrows in the world, CO2 moving in one direction, bicarbonate moving in another direction, it must be a mixed disorder for sure. Suppose if you're having a primary metabolic disorder, then which means this is second rule number 2. You should always expect the CO2 to change because it's fast. It should change in a primary metabolic disorder as soon as the event happens. But if it does not change, it remains normal. That indicates a mixed disorder. Similarly, in a primary respiratory disorder, if it's acute, it should have a normal bicarbonate only. If it's chronic, they should have a changed bicarbonate, but of course in the same direction. Similarly, same thing can be extrapolated to rule number 4 in a primary respiratory disorder. There should be a change in the CO2. If there should be a change in the bicarbonate. There should be a normal bicarbonate in acute respiratory disorder. If you don't have a normal bicarbonate, you have a grossly changed bicarbonate, then it becomes a mixed disorder. Similarly, in a chronic respiratory disorder, you should expect a change in bicarbonate. But if you don't see that, and if it's a normal bicarbonate, indicates again a mixed disorder. These are the 4 quintessential rules that are very important. More than 90% of the questions can be answered just based on these 4 rules. Nothing else is needed. But still, you have a long way to go as far as acid-base problems are concerned. But I believe that with this tricks itself, more than 90% of the problems can be solved so easily. Not a big deal at all. I'll tell you the examples after some time. So how you can solve these problems very easily. Meanwhile, if you have any doubts, you have any problems, you can keep it ready so that as we move on, I'll discuss your problems also. Maybe it can be a two-way interactive session. You can keep on discussing problems after problems. So once again, I'll tell the 4 rules. Arrows moving in opposite direction, 100% without a doubt, it's mixed. In a primary metabolic disorder, if there is no change in the carbon dioxide, it's mixed disorder because you always should have a change in the carbon dioxide if it's uncomplicated single disorder because it's fast. And similarly, in acute respiratory disorder, normal bicarbonate is quite common that there won't be much change because they'll be uncompensated. A chronic respiratory disorder, a changed bicarbonate is what you expect because there should be a compensation in a chronic respiratory disorder. On the contrary, in an acute respiratory disorder, if you have a grossly changed bicarbonate, that's something you don't expect. So that's a mixed disorder. In a chronic respiratory disorder, if you have a normal bicarbonate, then again, that's something you don't expect. So that's why it's a chronic disorder. I mean, it's a mixed disorder. So these are the 4 basic rules. So if you have understood these 4 basic rules and the equation, whatever I have told, these are going to form the basis of our future understanding. Shall we move on to the next area? Okay, we shall move on to the next one. So before going into the types and seeing the classification of acidosis, alkalosis, and all this stuff, it's more important to know about one particular thing that's called anion gap. That's very important. So anion gap is something very important ABG. Even if the entire ABG is normal, you should measure the anion gap. So the concept of anion gap is very simple. You have the number of cations in the body, and you're going to see the number of anions. So cations minus anions is what we call it as anion gap. Normal anion gap, we know it's less than 12. That's a normal anion gap. So the normal anion gap tells you one important thing. So that it tells you falsely, but that's not true thing. Falsely tells you that there are more cations than the anions. That's what it tells you. So then only you can get a positive value of more, around 12. That's a normal anion gap. So it falsely tells you the fact that number of cations are more than anions, but it's not the case. Always cations must be equal to anions. This is the rule. Cations should always must be equal to anions in the body, which means the net difference is basically 0. Otherwise, if it's not 0, if you have more charges in your body, then even a simple electrical current should make you attract towards it, and you will, anywhere you go outside, you will be stuck into the transformers and poles like a lizard. That doesn't happen. So you don't have any net charge. You don't produce any net charge in the body. Always your net charge is equal to 0, which means cations is equal to anions in your body. But this anion gap gives you an impression that cations are more than the anions. So which means why you have an anion gap in the first place? Because these are some unmeasured anions that are there. What are these unmeasured anions? These are basically organic anions. That's all. There are some anions you don't measure. For example, lactate. Then you have salicylate. Then you have other protein anions. Even phosphate is an anion. PO4 is there. That is also an anion. So many protein substances are there which can behave as anions. So you are not actually measuring certain anions. And because you don't measure certain anions, that is the reason why you have some gap between them, cations and anions, because you don't measure. It's our laboratory error. But we are going to make use of that laboratory error. So whenever I tell anion gap, it's a measure of unmeasured anions. So these are nothing but organic anions that are there. But if you want to know real definition of anion gap, it's a measure of unmeasured cations as well as anions. I don't want to confuse you, but that's the real definition. Because in the anionic side, you have missed a lot of anions. You have missed phosphate. You have missed your other anions like organic anions. You have missed certain protein anions. So you have missed a lot of anions here, nevertheless. On the other hand, if you see on this side, you have missed a lot of cations as well. You have missed potassium in this equation. You have missed calcium in this equation. You have missed magnesium in this equation. Lithium, if it's there. So many cations you have missed in this side. So which means ideal definition of anion gap should be a measure of unmeasured anions as well as cations. That is the ideal definition, because you have missed a lot of things in that. So why you have a gap between them? Because you have not measured some cations and you have not measured some anions. So which means that value is actually wrong. So that's the actual definition of anion gap. It's a measure of unmeasured cations as well as anions. But on the other hand, for our routine understanding, anion gap is not concerned with, like your cations. It's actually considered anions. That's why we call it as an anion gap in the first place. Which means what is more important in clinical practice is this unmeasured anions. That's what we want, especially the organic anions. So I can tell very easily whenever anion gap is increasing, I can tell my unmeasured anions are increasing. What are these unmeasured anions? My organic anions are increasing. That's all. Whenever anion gap is normal, which means it's very clear, my unmeasured anions are normal, which means it's not going too much, which means my organic anions are normal. There's no excess organic anions. That's what it means. So this is the basic concept of an anion gap. So organic anions increasing in, increasing anion gap, and organic anions are normal in the setting of a normal anion gap. So let us see a normal ion gap metabolic acidosis. Suppose if you see what are the examples of a normal ion gap metabolic acidosis, so that is called as NAGMA. NAGMA stands for normal anion gap metabolic acidosis. This is also referred to as a non-gap metabolic acidosis. So whenever they tell something called as a non-gap metabolic acidosis, you should not get confused by this terminologies. Non-gap metabolic acidosis means it's a normal anion gap metabolic acidosis only. So if somebody asks you what are the causes of a non-gap or a normal anion gap metabolic acidosis, usually I used to give by a mnemonic called URAT, but there are many other mnemonics are there available in market. So I used to give by a mnemonic called URAT. So U stands for ureteric diversion. Ureteric diversion. I'll tell you what is the problem in ureteric diversion. R stands for renal tubular acidosis. It could be a proximal or distal. A stands for Addison disease. And you can remember ammonium chloride poisoning also. So if at all you want, you can remember ammonium chloride poisoning also. And D stands for diarrhea. D stands for diarrhea. So this is the problem here. And diarrhea plus many other GI causes like fistulas, then you can think about other GI causes like pancreatic fistula, enterocutaneous fistula. So whenever there is some movement of gastrointestinal tract fluid somewhere else, you might result in the development of normal anion gap metabolic acidosis. First of all, you need to know why you get a normal anion gap metabolic acidosis in the first place, because here there is no rise in the number of anions, organic anions. So what's really happening here? So anion gap is sodium minus bicarbonate plus chloride. We know that. So this is the normal anion gap to start with. This is the formula for anion gap. So what do you mean by normal anion gap? Let us take sodium 140 as an average, bicarbonate 25 as an average, chloride as 105 as an average. So in this setting, if you take the anion gap will be 10. So that is a normal anion gap. So this is, let us assume a normal patient, normal person. So in a NAGMA, normal anion gap metabolic acidosis, due to some reason, the bicarbonate will be lowered. Due to some reason, bicarbonate will be low. So for example, take an Addison disease. What happens in Addison disease? You know that aldosterone is the one that takes up sodium, and aldosterone is the one that gives potassium. And this potassium that is coming down is the one that helps in pushing out the H+ as well. So and all these three things are basically happening with the help of aldosterone. Suppose if you do not have aldosterone in sufficient amounts in the body, then your sodium absorption might decrease and potassium excretion might decrease, and your H+ excretion might also hampered a lot. So which means this patient is actually having reduced amount of H+ excretion. So in the blood, the H+ concentrations are going to increase. This H+ concentration is going to reduce the bicarbonate. This H+ concentration is going to reduce the bicarbonate for sure. So if if the bicarbonate concentrations are reduced, and of course to keep the anion gap normal, to maintain the anion gap normal, because the amount of negative charges in the body is reducing, to maintain that loss of negative charges in the body tries to compensate by increasing the chloride. So once they increase the chloride, so they can maintain the number of negative charges, so the anion gap will be maintained normal. This is the basic principle of a normal anion gap metabolic acidosis. Suppose if you see this equation, example 140 minus bicarbonate, let us assume it is 15 here, and you are increasing the chloride to 115, because this change in the bicarbonate is compensated by the change in the chloride. Now it becomes 10 again, so you have a normal anion gap metabolic acidosis. So why it's called acidosis? Because your bicarbonate is low. So why the anion gap is maintained normal? Because you are increasing the chloride somehow. And this chloride increase can be absolute, or this chloride increase can be relative also. For example, a patient who's having a sodium of only 130 can have a chloride of 105. So even in this setting, you have a low bicarbonate, but chloride is here also will be increasing for sure, appropriately increasing. Because the sodium is low, the chloride increase will not be too much, thereby maintaining the normal anion gap, that is 10. So you cannot tell absolute increase in the chloride increase can be relative also. So nevertheless, the main issue here is low bicarbonate, but the chloride level will be balancing it out, this low bicarbonate, maintaining the anion gap in the normal range. So that's why we call it as a normal anion gap metabolic acidosis. That's what is happening in the setting of a Addison disease. Let us see what will happen in a diarrhea. In a diarrhea is quite simple. You are going to have bicarbonate loss in a diarrhea, because the pancreatic secretions and intestinal secretions are basically rich in bicarbonate. So when you have a diarrhea, you are losing a lot of bicarbonate out of the system. Once you lose a lot of bicarbonate out of the system, the body's bicarbonate will be low. And of course, if the body's bicarbonate is low, the body tends to avoid this kind of low number of negative charge in the body by increasing another negative charge in the form of chloride, which is a very important negative charge. So you can assume that even in diarrhea, the same thing happens, that the anion gap will be maintained in the normal range. But you can, ask one question, then what will happen in a high anion gap metabolic acidosis? Why we call it as a HAGMA in the first place? But before that, these are the causes of normal anion gap metabolic acidosis. That's what you need to know. So that is ureteric diversion, renal tubular acidosis, addisons and ammonium chloride ingestion, diarrhea and other gastrointestinal causes. Plus, apart from that, you have some other causes of normal anion gap metabolic acidosis, like for example, conditions that produce bicarbonaturia, like astazolamide can produce this kind of issue. And other toxins like toluene can produce this kind of an issue. And even very early renal failure can produce this kind of an issue. Very early renal failure. So why in early renal failure? In early renal failure, if you see, the main problem will be ammoniogenesis. We are talking about ammoniogenesis for quite a long time here. The ammoniogenesis will be impaired to a great extent if you talk about a early renal failure. Because ammoniogenesis is impaired in early renal failure, if this ammoniogenesis is impaired, then in this situation, you will not be able to trap this H plus from the distal nephron. If you cannot trap this H plus from the distal nephron, this H plus secretion will take a huge hit. If H plus secretion takes a huge hit, H plus starts accumulating in the body, and that's what we call it as a acidosis in the first place. Fine. That's all. Which means in a normal anion gap metabolic acidosis, you are basically not seeing any excess in any organic anions, which means the organic anions are still remaining normal, which means lactate, salicylate, and these kind of organic anions are still remaining normal. It's basically, you can imagine like a sort of a pure increase in H plus. That's what is happening in an AGMA. So because of some reason, there is either a pure increase in H plus or a pure loss of bicarbonate. It's only these compounds that are problematic here. And there is no problem in the organic anions. And because organic anions are normal, only the changes are happening in H plus, chloride and all these things. So that is why you are resulting in a normal anion gap metabolic acidosis. These are some of the important causes of normal anion gap metabolic acidosis. Very, very important for exams. In RT, you can assume. In renal tubular acidosis, I'll talk about that in some time. What is renal tubular acidosis? In renal tubular acidosis, either you will have a poor excretion of H plus, or you will lose bicarbonate too much in the urine. Losing too much bicarbonate in the urine is called proximal RTA. If you don't secrete H plus properly from distal nephron is what we refer to as a distal RTA. So whatever may be the case here also, the problem is either you lose a bicarbonate or you gain a H plus. It's pure. There is no net increase in the organic anions in the body. Point number one, very, very important point. So which means organic anions are still normal, and you can assume that is the reason why anion gap is still normal. There is no increase in the anion gap. As far as this increased organic anions, which means high anion gap metabolic acidosis are there. After acidosis, I talked about toluene here. And it can be caused by other drugs like sevelamer. It can be caused by even cholestyramine. Cholestyramine also creates a situation sort of a diarrhea kind of a situation. You can remember cholestyramine like a diarrhea. Even that can produce this kind of NAGMA. But nevertheless, so these are the basic principles that cause NAGMA, that is normal anion gap metabolic acidosis. On the other hand, if you take a HAGMA, that is high anion gap metabolic acidosis, when you write a HAGMA, here the problem is very simple. The sodium minus bicarbonate plus chloride is the formula for anion gap. If you take this formula, and here you know very well, let us assume one of the important example of HAGMA is lactic acidosis. Lactic acid is an organic anion. So I can split lactic acid into lactate anion and H plus. Lactate is an anion and H plus is there. So this H plus that is given by lactic acid is the one that is going to reduce the bicarbonate, and that's what we refer to as acidosis. And this is the reason for acidosis in elevated lactic acid. The reason for acidosis. Now, what will happen in a lactic acidosis? Simple example. Now let us keep the sodium constant 140. And because the H plus is coming more from the lactic acid, my bicarbonate will be low. So let us assume bicarbonate is 15. But to be honest, there is no need for the chloride to change here. Why there is no need for the chloride to change? Because there is already a lactate anion that is compensating for decrease in the bicarbonate anion, which means, for example, this is not exactly true, but for one molecule decrease in the bicarbonate, there is already one molecule increase in the lactate anion, which means the number of negative charges that are decreasing is already compensated by the excess lactate anion. So there is no need for the chloride to compensate here. So I can assume chloride is still 105. Now if 140 minus in this condition, if you take it is 120 is equal to 20. Now you encounter a HAGMA, that is increased anion gap. So that is because you can imagine that there is some unmeasured anion in the form of lactate here that is actually increasing in this patient. And that is why my anion gap is actually rising. So which means the basic definition is increase in the unmeasured anions is going to increase the anion gap. Here the lactate anion is unmeasured. Because it's increasing, you're having a increased anion gap in that setting. So what do you mean by that? So now what are the causes of NAGMA? So as far as NAGMA is, HAGMA is concerned, you usually give a mnemonic called Goldmark. Even though there are many other mnemonic like mud pilers, mud pile, so many mnemonic are there, but I like this mnemonic, Goldmark. This is a mnemonic I generally tend to give. So in a high NCAP metabolic acidosis, G stands for glycols. So which means you're talking about ethylene glycol, propylene glycol. So these are the glycols we are talking about. So ethylene glycol and propylene glycol. So G stands for glycols, and O stands for oxoproline metabolite, which usually comes in setting of acetaminophen poisoning or paracetamol poisoning. Paracetamol poisoning. Oxoproline metabolite of acetaminophen poisoning. Then you can get with lactic acidosis. L stands for L-lactic acidosis. L-lactic acidosis. D stands for D-lactic acidosis. D stands for D-lactic acidosis. M stands for methanol. A stands for aspirin or salicylic acid poisoning. R stands for renal failure, especially the renal failure you're talking about is the late renal failure where uremic toxins will increase, and because of that your anion gap will be high. And the K stands for ketoacidosis. That is ketoacidosis, you have three causes basically: diabetes, starvation, alcohol, severe starvation. All these things can produce ketoacidosis. This is the three principal cause of ketoacidosis: diabetes causing DK, starvation, and alcohol. Clear? So these are basically the causes of HAGMA, that is high NCAP metabolic acidosis. So here you will be seeing two lactic acid. One is D-lactic acid and second is L-lactic acid. Of course, L-lactic acid is the most common form of lactic acidosis, even though D-lactic acid can form in the setting of something called a short bowel syndrome. Whenever they talk about D-lactic acid, always think about a short bowel syndrome. In exam, if somebody gives you a question of having a massive bowel resection, after that in the second day or third day, the patient comes with altered sensorium, it will be usually due to D-lactic acid. Because the short bowel syndrome, the carbohydrates will be undigested and coming directly to the distal part of the intestine where there will be so much of bacteria, they will metabolize this carbohydrate D-lactic acid, and that can mix in the blood causing this neurological problems. Usually it's a typical complication of a short bowel syndrome. Apart from the D-lactic acidosis, extremely rare to get in real life setting. And remember, this D-lactic acid, one important, problem. I can write D-lactic acid here also. Because D-lactic acidosis, if you take a lot of textbooks, they'll be given in both normal NCAP metabolic acidosis as well as high NCAP metabolic acidosis. That is because of one important reason. Because if this anions, what you're talking about, this organic anions, is absorbed actively by the kidney, or if it is not absorbed by the kidney, if it is absorbed rapidly by the kidney, or if it is if it's not secreted by the kidney, then they will start getting accumulated in the body and they will result in all this HAGMA and all. Suppose if there is a anion that can get eliminated faster, like the kidneys can secrete that anion, and or if it can be filtered by the glomerulus very fast, and if it can be excreted out of the system so fast, then once you eliminate the anion, there is no point in having this kind of increased anion gap. So D-lactic acid is one example where it can be eliminated faster and it can be pushed out of the kidney, can be secreted from the tubules directly so that it will get eliminated faster. So that is the reason some textbooks will give it can produce a normal NCAP metabolic acidosis. But that is quite confusing thing. D-lactic acid is something that can be there in both NAGMA as well as HAGMA causes. Again, it depends on the clinical background. For me, any acidosis in a patient with a short bowel syndrome, whether it is NAGMA or HAGMA, I will take it as a D-lactic acidosis only. So clinical context is more important rather than just mugging up these causes. But nevertheless, renal failure also, if it's early, they can result in NAGMA. If it is late, they can result in HAGMA. So which means there will be a lot of overlap between these causes. So don't try to just mug up these causes. See the clinical background of what is really happening in that particular patient. So be careful in that. So why kidney failure early, it will produce only normal iron gap metabolic acidosis? Because it's a problem of ammonia purely. They cannot produce ammoniogenesis. That's the problem. As the kidney failure progresses, the GFR will be affected worst, and these toxic anions will start getting accumulated in the body, then result in the development of high iron gap metabolic acidosis. Remember, always CKD will have HAGMA. AK in very early stages will have NAGMA. AK again, if it progresses to a little late stage, it will result in development of HAGMA only. So don't get confused by that. It's very important thing. In exam, kidney failure means always HAGMA only. Don't take it as NAGMA. Only for your understanding, I told you. Only to complete, I told you. But remember, in exam, kidney failure always means it's HAGMA only. And lactic acidosis always means it is HAGMA again. D-lactate may be NAGMA or HAGMA, but the clinical context there is more important to diagnose a acid-base disorder. So this is the concept of blood anion gap. That is anion gap metabolic acidosis. Clear? So which means whenever the patient is having a metabolic acidosis, this becomes very, very important, whether it's a high anion gap or if it's a low anion gap. Clear? So then as far as the high anion gap metabolic acidosis is concerned, whenever you know that the patient is having a high anion gap, now finding out something called a delta-delta ratio becomes very, very important. Whenever you encounter a patient with the HAGMA, or whenever the patient is having a high anion gap for any reason, it's always important to find out the delta-delta ratio. So what do you mean by delta-delta ratio? So which means here we call it as the delta anion gap divided by the delta bicarbonate. Now you will be confused more. So what do you mean by delta-delta ratio if the patient is having a high anion gap? So it's actually quite simple. So understanding is very simple. Suppose, let us assume in a high anion gap metabolic acidosis, let us assume a lactic acidosis. You'll be understanding very easily based on lactic acidosis. So that's why I'm telling lactic acidosis first. So let us assume one molecule of lactic acid is increasing. So this lactic acid is going to produce one molecule of H+. Let us assume one molecule of lactic acid. One molecule of lactic acid is there. This lactic acid is going to produce one molecule of H+. This one molecule of H+ is going to neutralize one molecule of bicarbonate. And of course, this one molecule of lactic acid is going to result in one molecule of lactate anion as well. So once lactic acid is split, it's going to produce one H+, that's going to neutralize one bicarbonate. So one bicarbonate will be low. And for the exact same thing, you're going to have a one increase in lactate anion. It's very clear, isn't it? Which means that's why, like the anion gap will be basically maintained. That's the idea. So this one molecule increase in lactate is the thing that's going to increase the anion gap, which means it's a measure of unmeasured anions. So one molecule increase in anion gap will be there. Or you can tell one unit increase in anion gap will be there. Which means, suppose if the normal anion gap is 12 for that matters, if anion gap becomes 15, we're talking about anion gap. Now let us assume because of lactate, anion gap becomes 15. And bicarbonate, we know it is 24 now. It will become 21. So for 3 increase in anion gap, there is a 3 drop in bicarbonate, which means most of the times, the increase in anion gap and the decrease in the bicarbonate, both will be almost equal, which means the change will be neutralized. So that in most of the scenarios, that should be equal to 1. That's what I'm trying to tell. The change in the anion gap must be equal to the change in the bicarbonate, because one increase in lactic acid, one H+ it's giving, one H+ neutralizing one bicarbonate. That one bicarbonate is actually equal to one increase in lactate anion. That one increase in lactate anion is going to be equal to one increase in the anion gap, which means that increase in anion gap, that one increase in anion gap is basically equal to one decrease in the bicarbonate. So the actually effective change, that one increase here in the anion gap and one decrease in the bicarbonate, is actually a sort of a neutral sort of a scenario there. So in that setting, it should be equal to 1. So that is the basic idea of this entire thing I'm trying to tell. That works very well for lactate, even though there are certain acids which may not produce exactly equal to 1, their neutralization will be different. So exactly one molecule of particular acid may not neutralize one molecule of bicarbonate, so that changes will be there. So normally this delta ratio, I'll take it as 1 to 2 as normal. This ratio of 1 to 2 is considered to be normal. Somewhere in 1 to 2 is typically considered to be normal. So how will we calculate delta ratio? Delta anion gap means I know normal anion gap is 12, so anion gap minus 12. In a setting of acidosis, my bicarbonate will be low. So 24 minus bicarbonate will tell you the delta ratio, which means I'm going to see the change in the anion gap and compare it to the change in the bicarbonate. If both are equal, then fine. So that's something expected. That's what should happen in a high anion gap metabolic acidosis. In any high anion gap metabolic acidosis, and the ratio being 1 to 2 is considered to be normal in most cases, because that exact neutralization will not be 1. For example, lactic acid, it's 1.6. So that's why it need not be necessarily equal to exactly 1. So, but 1 to 2 is what we consider to be normal in case. In this particular setting, suppose if the ratio is less than 1, or the ratio is more than 2. So this is where things get complicated. Suppose if the ratio is more than 2 in this setting, I can tell that this patient is having associated metabolic alkalosis. Now you will be completely spinning your heads off. I'll tell now there is an associated metabolic alkalosis. If the ratio is less than 1, I can tell there is an associated NAGMA. Now you will be asking 2 questions. Sir, why are you torturing us? You told it's a iron gap metabolic acidosis. Now you're telling associated non-iron gap metabolic acidosis. And body, yes, it works like that. It's not a machine. For example, a patient who's having a CKD will have a HAGMA. Let us assume this patient is having a vomiting now, continuous vomiting. Because he's having a continuous vomiting, he will develop a new metabolic disorder because vomiting results in development of metabolic alkalosis. Or this CKD patient can develop a diarrhea. Because he develops a diarrhea, he will develop a non-iron gap metabolic acidosis. So which means HAGMA need not be associated only with your HAGMA alone. It can be having associated metabolic alkalosis, or it can be having associated NAGMA also, non-iron gap metabolic acidosis also. How can you find out? It's based on the relative changes in the bicarbonate and the anion gap. So let us assume one important thing. Suppose in CKD, there is some organic anion that is increasing the anion gap. Anion gap is increased probably to 20, let us assume. So baseline bicarbonate is exactly decreased to that of the anion gap. So that is anion gap is increased by 8 from a matter of 12. From 12 it is increased to 8 units. So let us assume bicarbonate here is 18, and that is appropriate. Why? I mean 16. Why bicarbonate of 16 is appropriate here? Why bicarbonate of 16 is appropriate here? Now if you see the delta ratio, 20 minus 12 divided by 24 minus 16 is equal to 8 divided by 8 is exactly equal to 1. So I can see this is a correct HAGMA. This is a HAGMA only. In this patient, I'm having only high iron gap metabolic acidosis. Suppose if this patient is developing a vomiting, let us assume this patient is developing a vomiting. Vomiting is an alkalotic condition. So definitely this patient is having a baseline 16 because of CKD. So now this patient is having only a CKD. Now what happens? Now because of vomiting, this bicarbonate will increase to a new value. So now maybe let us assume bicarbonate has become 22 because of the vomiting. Now bicarbonate has become 22 because of the vomiting. Now if the bicarbonate has become 22 because of the vomiting, this is not correct and this is not appropriate for the HAGMA. I mean, it's not one thing. There is 2 things that is there now. Now if it is say the same ratio, the anion gap is still 20 minus 12. Now the bicarbonate has become 22 now. Now if it is the ratio 8 divided by 4 is equal to, not 8 divided by 4, 8 divided by 2 is equal to 4. So that is why I told you, whenever the patient is having a delta ratio of more than 2, now I can assume there is some new event that has happened and that has raised the bicarbonate to a more value than what it should be. So which means there should be that raised bicarbonate from the baseline value must be due to metabolic alkalosis. So that is why whenever the delta ratio is more than 2, I can tell that the patient is having a associated metabolic alkalosis, not only having a HAGMA. So this is a very important scenario. Or let us assume this patient has developed a diarrhea now. Now suppose this patient has developed diarrhea in the sense, the baseline bicarbonate of the CKD patient is 16 and the anion gap is 20. Now diarrhea should have reduced the bicarbonate to a new value because diarrhea does not change the anion gap. It's only going to change the bicarbonate. That's why I told you pure decrease in bicarbonate or pure increase in H+. Now if the same patient is getting a diarrhea, now what happens is the anion gap remains the same. It's 20 because diarrhea is not going to change the anion gap. Now, as far as the bicarbonate is concerned, now the new bicarbonate would have become 10 because of the diarrhea he has got a little bit more decline in the bicarbonate. So now it has become 10. Now if you see the delta ratio, 20 minus 12 divided by 24 minus 10 is equal to 8 by 14. Now definitely the ratio is less than 1. So that is why whenever the ratio is less than 1, I can tell that this patient is not only having HAGMA, there is another process that is reducing the bicarbonate only compared to the anion gap. If it reduces the bicarbonate only, means there should be an associated non-anion gap metabolic acidosis. That's why, I mean, even if you understand this or not, I don't have a problem. But the most important thing is to understand the delta ratio. Whenever you have a HAGMA, or whenever you have a high anion gap in the patient, it's very important that you see the delta ratio. If the delta ratio is more than 2, then you call it as a associated metabolic alkalosis. If delta-delta ratio is less than 1, you call it the patient is having associated NAGMA, not only the HAGMA in the background. Do you understand this or not? So that I can move on to the next one. Clear? So this is what we refer to as a delta-delta ratio, which is very important, especially in the setting of a high and gap metabolic acidosis. But on the other hand, if you think about a normal and gap metabolic acidosis, one more equation is very important in normal and gap metabolic acidosis, that is urinary anion gap. Whenever you think about a NAGMA, that is normal and gap metabolic acidosis, the next thing you have to look for, even though we don't see most of the times because the cause will be obvious, it's better to see the urinary anion gap. So if somebody asks you what will be the formula for urinary anion gap is equal to urine sodium plus potassium. This is a standardized formula minus urinary chloride. This will be the urinary anion gap. So remember, the urinary anion gap is also the same thing. So urinary anion gap means whether the unmeasured anions versus cations, which is more and which is high. That's what we measure using the urinary anion gap. So urinary anion gap can be negative or urinary anion gap can be positive. So based on that, basically urinary anion gap should be 0. That's the whole idea. But we can, in the setting of a normal anion gap metabolic acidosis, you can assume whether it is a negative or whether it's a positive urinary anion gap. Based on that, you can give some causes. So this chloride, we know, is actually representing the ammonium. This chloride is basically representing ammonium, nothing else. It's just representing the ammonium. Suppose if chloride is more means ammonia genesis is more, which means my anion gap has become negative. That's what it means. My ammonia genesis is more and anion gap has become negative. So what are the conditions that produce negative anion gap? Typically the GIT causes, the GIT cause of NAGMA. What are the GIT cause of NAGMA? This ureteric diversion, then this diarrhea and other gastrointestinal causes like pancreatic problems, fistula. All these things are basically GI causes. Why ureteric diversion is a GI cause? Because ureteric diversion, where you divert? You basically divert to the ileum only. So now currently diverting to the ileum is the most common procedure. Previously they have been doing to the sigmoid, called as ureterosigmoidostomy. Now currently they divert to the ileum. So why diverting to the ileum is a problem? Because in ileum there will be more chloride absorption that is present in the urine. That more chloride can result in NAGMA, normal anion gap metabolic acidosis. We have studied that already. More chloride is always a problem. So that's why ureteric diversion is also like a GI cause only. So these are the usual GIT cause, which is going to produce a negative anion gap. As far as positive anion gap is concerned, which means the anion gap is high, means usually it indicates a renal cause. More often, renal cause means we are talking about a renal tubular acidosis, and probably we can talk about an Addison disease. So these are some classical conditions which produce a positive urinary anion gap. But in the RTA itself, you have two types of RTA, distal RTA versus proximal RTA. Distal RTA is the one that consistently causes a positive urinary anion gap, whereas rarely proximal RTA can produce both positive anion gap as well as a negative anion gap, depending on the situation that we are encountering. That's the idea here. That is the idea. Clear? So now we can ask why in the setting of a GIT cause, let us assume a simple GIT cause. To understand, to get a grasp of this, we need a simple GI cause, that is diarrhea. Why in diarrhea you should have a negative urinary anion gap in the first place? Which means in diarrhea you should have a high urinary chloride excretion. Because there is a high urinary chloride excretion, you should have a reduced anion gap. Because this value is getting higher, urinary anion gap is getting lower. So that is our assumption. So why urinary chloride should be high, and why in a diarrhea your like ammonia genesis should increase? It's quite simple, because diarrhea produces loss of bicarbonate. Loss of bicarbonate has to be, means there will be increase in H plus in the body. Both are like white and black. So both are like North Pole and South Pole. So one is increasing means other should decrease. So loss of bicarbonate I can equate to gain in the H plus in the body. This H plus has to be got rid of. To get rid of this H plus, of course, the only way to increase the ammonia genesis, this ammonium should always trap a lot of chloride. So that's why I told you in the beginning itself, ammonium can be easily assessed approximately by seeing the chloride. So whenever this H plus has to be increased in amount, so this chloride has to be increased. So that's why your chloride values are increasing. That is why GIT causes typically tend to produce a, like negative anion gap. Fine. On the other hand, if you let us take a distal renal tubular acidosis, as far as the distal renal tubular acidosis is concerned, so the problem is H plus secretion. This is not happening. That's what the problem is. H plus is still getting accumulated here in a distal renal tubular acidosis. If it's getting accumulated here, now what will happen? Even if the proximal tubule produces sufficient amount of ammonia, it doesn't matter. They cannot come and get eliminated, because only if this H plus comes out, you can anyway combine with ammonia. So your ammonium excretion will be so low, which means your chloride excretion also will be low. Because of that, your chloride will drop. Because chloride is dropping in this equation, the urinary anion gap should increase as a rule. So that is why in distal RTA especially, urinary anion gap is increasing, to be honest. Clear? So that is why I used, but proximal RTA can produce anything. Proximal RTA can have a normal anion gap, or proximal RTA can have a increased anion gap also. It depends. Like it depends on a lot of other entities about the function of the distal nephron. The distal nephron is still functioning normally. In proximal RTA, the urinary anion gap will be negative. Negative only. If the distal nephron is not able to produce sufficient amount of H plus, then urinary anion gap can be positive also. Proximal RTA is something different, but nevertheless, GAT causes are more important here, and RTA, especially the distal RTA, is the most important here. And Addison, same principle. In Addison, if you have low aldosterone, you will not be able to eliminate the H plus out of the system. So that is why this is happening. So there is a simple mnemonic for this. To understand, there is something called a negative. That's how you can remember. So I can tell the GAT cause, the gut causes usually will have a negative anion gap. Clear? So it's very easy to understand. So whenever you see, this urinary anion gap is usually assessed only in the setting of a NAGMA, non-anion gap metabolic acidosis. So if you see the urinary anion gap to be negative, you can think about a GAT cause like a diarrhea, or probably a pancreatic problem, or a fistula kind of a reason, or probably sometimes can be a proximal RTA. If the urinary anion gap is positive, usually it's a renal failure. Renal cause, that is renal tubular acidosis, which is resulting in the setting of a probably due to Addison disease or a proximal RTA. So let me ask you one easy question. Let us talk about early renal failure. Early renal failure is going to produce, like normal anion gap metabolic acidosis. This is just to understand, but in exam, renal failure is HAGMA only, but early renal failure typically results in NAGMA only in most of the clinical scenarios. What will be the urinary anion gap if early renal failure is producing a NAGMA? What will be the urinary anion gap? It will be positive or negative? It will increased or decreased? That's what my question is. If you understood this, this should be easy for you. Negative or positive? I'm getting mixed answers. I don't know what is the confusion here. Okay, let me ask you another question. So what will happen if in an early renal failure, what is the primary physiology there? Why you get an acidosis? What is the problem I told you? You're going to type and tell. So what is the problem I told you that happens in early renal failure to result in acidosis? Low ammonia production. Yes, impaired ammonogen is the main reason for early renal failure that is causing acidosis. If there is low ammonia production, so what will be the chloride excretion? It will be low or high? Chloride excretion will be low or high? Chloride excretion will be, of course, low. If the chloride excretion is low because of low ammonia production in early renal failure, what will be the urinary anion gap? It will become positive or negative according to the equation. Of course, yes, it will become positive. That's why you can write, that's why I told you, as a whole, renal causes generally tend to produce a positive anion gap, and GAT causes tend to produce a negative anion gap. I told you the mnemonic also, that is negative, NEGUTIV, negative. I think this must settle the debate. So in future, whenever you see an anion gap sort of a thing, whenever there is a HAGMA, that is high anion gap metabolic acidosis, always see the delta-delta ratio. Whenever you have a NAGMA, that is normal anion gap metabolic acidosis, always look for the urinary anion gap if your data is correct. If your data is okay, then you can go for a normal anion gap metabolic acidosis also. Okay, before going into the steps of the acid base, and next session will be based on renal tubular acidosis and steps in acid base gas analysis. So before going into that, you can go for a break and come back at 5 o'clock. I'll start by 5 o'clock. You can go for a break now because it's been a long session. So think you don't have any doubts in this particular session. So stay tuned. Next session is going to be another power packed one, and we're going to discuss a lot of other entities also. Go back to your break and come at 5 o'clock. I'll start by 5. I think the connection will be better today because morning I've tried people to come and do something. So they have done something. The problem is in the tree and the ant that is coming into the house. That is where the problem, it seems. That's what they found out. Let us see. Okay, bye-bye.